Everybody, and thanks for showing up for our first session. We have a few brief words and of introduction from, well, I'll let you go ahead, but we're very happy everybody's here, and we hope to have a very nice thing today.
Hi everybody, good morning. I'm happy you all made here after last night. And just introduce you, IMPA's director, Antônia Franco, Dr. Antônia Franco. Also with us, IMPA's vice director, Dr. Ilanja, sitting in the front row. And just to officially open the workshop, I will pass the microphone to Dr. Antônia Franco to say a few words of welcoming for you. And yeah, that's it. I hope you are enjoying. Professor.
Good morning. Guten Morgen. So I'm going to read something here, okay? So first will be greetings to all who are present here and the ATTO workshops. Talking about IMPA can be summarized in words that involved our mission, which is to generate and disseminate knowledge, technologies and innovations, and train people to contribute to the formulation of public policies and actions for the sustainable development of the Amazon. Today, when I woke up, I thought about what I could say in this opening of the workshop. I was thinking about this. What can I talking for all? So, talking about the importance of studies related to climate change, the ATTO Tower as an instrument of these studies. The need to involve a greater number of researchers and how much this possibility of mobility between studies and research from different countries is necessary for this integration. For me, ATTO is an integration of many persons from the world. This is so important for the science. I summarize all these welcome speeches in a message. Study, work for the planet, and when challenging yourself, remember that the greatest challenge of science is its ability to understand and to transform. Welcome and much success and discoveries for all. Thanks for staying at IMPA, and thanks for the science. Thanks so much.
Thank you. Thank you. You're right. Straight to the science then. Who is the next speaker?
It's open.
Yeah, it's open.
The conference is now open, and I guess the first two speakers are, we go into the greenhouse gas session. Oh, no.
No.
Viviana will now tell us what we do next.
Okay, sorry for the confusion. But we have two plenary talks today in the morning. We have Dr. Luana Basso and Dr. Freshly graduated Dr. Santiago Botilla. So I think the first presentation from Dr. Luana Basso, please.
So good morning, everybody. It's a pleasure to be here. I'm not working with ATTO, so I will talk about methane emissions in a more regional perspective in Amazonia. I'm from INPE. I finished my postdoc last week in the Luciana's GAT group, and I will present the results that we published last year. It's about 9 years of methane emissions in Amazonia. And we did this study with vertical profile with aircraft measurements. So it's a bit different from ATTO, but then Santiago will give a more local perspective after. So, could you, oh, next one. Okay. It's not working. Okay. Just a detail. Okay. Just to quick, briefly introduction about methane emissions and why we are studying this and why it's so important. In general, in the planet, we know that methane could be emitted by anthropogenic or natural sources. In a global perspective, we have now around 60% of emissions from anthropogenic sources, but we also have natural sources as like wetlands, dominating almost 40% of the emissions. And we also have some sinks from the atmosphere, but this is mainly driven by the reaction with the OH radical. And why is it important we study methane? And in these last years, this is a big question from the scientific community, because we are seeing a very significant increase in the last years after a first period that we have more stability in the levels, in the atmospheric levels between 2000 and 2006. We start to see again that the levels in the planet is increasing. And more recently, after 2014, this is being accelerated. And what is driving this increase in the emissions is a very big question that no one knows exactly yet. And one question could be if this is just driven by anthropogenic emissions or if it's now a climate feedback from the climate change, like increasing temperature and the change in precipitation and a response from the wetlands. But why Amazon is important in this scenario is because we know that wetlands are the main natural methane source to the atmosphere, and the tropical regions that have the largest areas of wetlands in the globe have a low regional samples, and this is changing a bit in the last years, but we know that we still have a large variation in the Amazon estimates, and almost 20% of the Amazon area is wetland, permanently or seasonality wetlands. And how Amazon is linked with the global perspective in the methane budget is still a question we are trying to answer. So this is one of the motivations of our project. This is a project led by Luciana Sgatti from INPE, São José dos Campos in Brazil. And we work with vertical profiles in the lower troposphere. So we have vertical profiles starting in 4.5 kilometers high and going down in different levels until the most safe altitude near to the canopy of the trees. We have measurements since 2010 in 4 different places in Amazonia. So we have 2 in the more east part of Amazonia and 2 in the west part, and 2 more in the north, near to the main rivers, and 2 more in the south part of Amazonia. So we feed this kind of distribution of the sites. We could have a regional resolution because we are having measurements with aircraft, so we are talking about a very large area of measurements. Also, because we have now more than 10 years, we can have temporal resolution, try to understand seasonality and the interannual variability and possible trends over time. And we use a kind of simple mass balance technique to estimate fluxes. I will talk very quickly about this and just give more details on the results. So we estimate flux based on the concentration of the vertical profile. So we have, in general, 12 or 17 different altitudes to construct these vertical profiles. And we need to remove the global concentration, because we are just interested in the difference between what the Amazon is contributing for source or sink. So we estimate the global concentration, like the background concentration, how much is the methane concentration in the air that is entered in the continent, based on a mathematical estimate with the NOAA surface sites. And this difference is divided by the time based on the back trajectories using a model from NOAA also, that is the high split model. When we do the vertical profiles, we are measuring methane, but also other gas, like we also have measurements with CO2 and N2O, and also we have measurements of carbon monoxide. So if you have carbon monoxide and methane being sampled and measured in the same time, in the same samples, we could try to estimate a ratio between CO and methane and estimate how much of the total flux that we are seeing in the vertical profiles come from fires. So it's a simple method for we try to know what kind of sources we are talking about. So for these estimates, we use this relation. So for the vertical profiles during the dry season, we could see very clear in the carbon monoxide the increase in the concentrations in the different altitudes. And this indicates when we have biomass burning happen. And then in some profiles, we could see very clear that the methane concentration have this similar behavior or the similar increase. So when we this comparison here, we could estimate how much of methane we have when we have amount of CO going to the atmosphere by fires. So we calculated this ratio for each of these sites. And so each region have a different relation between CO and methane. And then we apply this to our CO flux to estimate how methane comes from fires. And as I told before, we are talking about vertical profiles that have a very large resolution in the measurements. So we need to understand better what kind of a region we are talking about, because we are measuring all process that is happening in this large area. So all sources and sinks that happen in all the region between the coast and the sample site is influenced these measurements. And then based on the back trajectories, we could calculate a simple influenced area. And then because we have much more influence of the region near to the vertical profile site, we could apply a weight, so we can give more importance to the process that is happening near to the site in relation with the process that is happen more frequently. So the colors here is just with more redness, we have more influence in what we are measuring. And with these regions, we can try to get another kind of data sets to understand better our emissions, like precipitation, temperature, and water availability, to try to understand what could be the seasonality and the interannual variability. So talking from the first results, we published this last year in a paper in Communications on the Earth and the Environment. And this is based on the results between 2010 and 2018. So we are talking about 9 years of around these 600 of vertical profiles in the Amazon, different places. And when we start the mean of the vertical profiles and the 9 years mean in black, we could see first when we compare the different altitudes, we can have an indication if these regions are source or sink of the gas that we are studying, so in this case of methane. When we compare the lower part of the vertical profile, around below 1.5 kilometers high, we're talking more about the process that happens in the region or more near from the forest, in comparison with the top of the vertical profile, that we have less influence from what is happening in the surface. So when we compare and see that the lower part of the vertical profile have higher concentrations than the top of the vertical profile, this suggests to us that the region is a source to the atmosphere. So the first indication of our measurements is just that the Amazon is a source of methane. We started to see a difference between the regions and the sites that we see. So when we look at this northeast part of Amazonia, near to Santarém, we could see that the increase in the concentrations is higher or larger than the other side, and that we could have a regional variability in emissions. And when we are talking about this, and because Amazonia is a large area and a very diverse ecosystem, and now we have a lot of disturbances happening in different levels, we need to be carefully to understand the difference between the regions. So based on this, we try to understand a bit better how these regions are different in disturbance levels or in climate conditions. And one of the analyses that we did is using the MAP biomass data to understand better what kind of anthropogenic activities we could have in these regions. So we found working with like quarterly areas, because we see seasonality in the air mass back trajectories. We understand that the southeast regions, like near to Alta Floresta and Rio Branco, we have large areas with agriculture use, of land use, and also high levels of deforestation, and also near to Santarém region in this northeast part. And then when we go to the west part, here we have two sites, because we started the measurements near Tabatinga until 2012, and then we need to move the site to Tefé because of logistical problems. But they are very similar in concentrations, and then we consider just one long time series. So we could find that this northwest part is less impacted by disturbances with deforestation and also with agriculture. And then thinking of the methane emissions, one important anthropogenic emissions come from the cattle, the enteric fermentation process. So to try to understand what we are seeing in the vertical profiles, we need to think that we have anthropogenic sources and natural sources. And we are not able with the vertical profiles to point how much come from each source, because we are not able to distinguish each process in the samples, but we could use another data sets to try to understand a bit more our budget. So we use the EDGAR database to try to investigate how much of our measurements or our concentration and fluxes come from anthropogenic emissions. This is a global data source, completely free available, and then we do this. We did this regional analysis for each of the influenced areas. And when we look at the anthropogenic emissions from EDGAR for these regions, we could see that the main activity that is emitting to the atmosphere in Amazon methane is coming from enteric fermentation. For some regions, it's almost 90% of the total anthropogenic emissions, and for other regions, this is a bit less. But in general, what we found is that for the south part of the Amazon, like a large influence of anthropogenic emissions, and mainly from cattle, mainly from enteric fermentation, representing in the southeast part almost 35% of all the emissions that is not coming from fires. Okay, this is fire emissions is still subtropical. So we have this 9 years of measurements, and then we have the possibility to understand the mean seasonal cycle of the emissions in the Amazon and try to compare and understand what these emissions. So the first thing that I would like to highlight here is that for Santarém region, this northeast part of Amazon, emissions in this region is like 3 times higher than what we are seeing in the rest of Amazonia. So just to be very clear that here we are seeing much more emissions than the other sites. And then when we look at the gray bars, we are talking about the total emissions, anthropogenic and natural process. When we look at the orange bars, we are talking about the fire emissions that is estimated based on the carbon monoxide that we have in the vertical profiles. So when we look at the fire emissions, we could see that in this east and south part of Amazonia, this is we have a large contribution from fires than in the northwest part, as expected, because we are more near disturbance areas and the arc of deforestation. And we can see a very clear seasonal cycle related with the dry season, higher emissions in the dry season, but in some the wet season, or in general the wet season, but we have more influence from the North Hemisphere during this part of the year in our regional area of influence. Then after we subtract the emissions from fires, in these dark blue bars we have the non-fire emissions. That is anthropogenic process, not related from fire, like the enteric fermentation, and also the wetlands. So we use the EDGAR database to subtract the anthropogenic emissions and consider the sink from soil from the literature. And then we estimate how much is the wetland emissions in this more, the other blue bar, okay? And we could see that from the non-fire emissions, we see that wetlands is driving these emissions, okay? So removing the fires, the mainly source to the atmosphere in Amazonia come from wetlands. When we go to the places more in the north of Amazonia or in the central north of Amazonia, we see that this is like more than 90% of the non-fire emissions. And when we go to the south part, this is a bit less because we have less influence from the larger rivers and less wetland areas, but we also have much more anthropogenic emissions in these places. So another important point to highlight is that we see variability in emissions in magnitude with higher emissions in northeast part, but also in the seasonality from the wetland or the non-fire emissions. In the northwest part, we also didn't see too much seasonality, clear pattern, because this region have lower variability during the dry and wet season. We also, in our region of influence, don't have a really dry season with precipitation less than 100 millimeters. But when we go to the south part of Amazonia, we could see very clear that the emissions make sense with the higher levels of the wetland. Here represented by the blue lines, that is based on GRACE measurements to try to estimate wetland areas. And when we go to this northeast part, then we have a kind of surprise because we have a different seasonal cycle with two important peaks in the emissions, one in the wet season, but before the peak of the wetlands, and in the beginning of the dry season with larger emissions similar to the wet season. And why this is happening is still an open-ended question. So we are trying to understand better what drives the emissions there and what is the source of this large emission also in the season. Then, looking these 9 years, we try to investigate interannual variability. So the scientific community of methane, someone found that we have an increase in emissions in Amazonia based on satellite data. That could be a reason for our partial region that we have an increase in emissions in the globe. But when we look at our measurements these 9 years, we didn't see any kind of trend, so we didn't see an increase in emissions in Amazonia. And also we see that during the hotter and dry years, we have large fire emissions, which was expected, like during the El Nino in 2015 and 16 and 2010. And when we look at the wetland emissions, we didn't see a clear relation with the climate conditions. So we cannot explain very well the interannual variability in from the wetlands. And then we compare our flux estimates from the vertical profiles with like wetland bottom-up models to try to see if we are seeing the same estimates for Amazonia. And in general, when we compare our sites, we have a good agreement between the different regions, but for this northeast region of Amazonia near Santarém, the models could not predict the same amount of methane to the atmosphere that we are seeing. So they also see higher emissions here, but this is not, or this is around 40% of what we are measuring. So what is happening in this region or why we have these large emissions is still another open question. So the northeast part of Amazonia is an open question for methane emissions. And when we just do general balance for the whole Amazonia between the two methods, our approach and the annual approach from wet charts, wetland emissions, that is a bottom-up estimates, we see an agreement, but we still need to think that regionally we have this discrepancy with the northeast part of Amazonia. And then just to start to summarize, when we compare our total emissions based on the vertical profiles for the Amazon region, we are seeing that this region is contributing with around 8% of the total emissions of methane. And we look for like the fire emissions that happens mainly in the tropics. We are seeing that for estimates, we are looking around 45% of the global emissions just happening in the by biomass burning in Amazonia. And when we look the wetland emissions, this contribute around with 23% of the global emissions. That also is mainly driven by the tropical regions that have these large areas. But when we look this regional variability, we see that in general, wetlands are the main source of methane to the atmosphere in the Amazon. It means that we cannot do, or we can just be not try to reduce or control this with the time, but this is not true because we have large emissions from fires, mainly in this south part of Amazonia, so we can try to reduce this, is almost 20% of the emissions. And we also have large emissions from anthropogenic sources, mainly in these disturbance areas in the south part of Amazonia. So this is the point that Brazil could try to reduce, thinking about this new agreement with the COP26. And then we also, just to highlight a bit more this regional discrepancy with the northeast part, this is a completely independent result based on satellite data and inverse modeling from Chris' group from University of Leeds. And using GOSAT data and the inverse modeling, he also found these large emissions in the northeast part of Amazonia. So they reproduce the same regional variability that we are seeing in the vertical profiles, what make more sense. And we also don't know why this is happening. And we also found a similar or a more similar result in the global, in the total budget for Amazonia. But one point that we need to highlight is that this study and other studies that use GOSAT data from satellite, are seeing a positive time trend in Amazonia, and with the we didn't see any kind of time trend. So this is another point that we need to investigate more, why we are seeing different results from satellite and the more regional measurements. And then just to summarize, so Amazonia is an important source of methane to the atmosphere, with mainly wetlands dominating these emissions, but we still have a large contribution from biomass burning that we could try. Based on the vertical profiles, we didn't find any kind of trend, increase or decrease or changing the emissions, in contrast with analysis based on GOSAT data. We also see clear seasonal patterns that can be partially explained by different climate controls, mainly in the northwest central region, that we see this constant week of seasonality that is constant with precipitation and wetland areas. In the south part, we see this link with the wetland water availability, the levels of wetlands, and the northeast part that have this peak is still a question. So we are trying to understand this, and we highlight that we have this east-west contrast in emissions with much more large emissions in the northeast part of the base, and we still know what is happening. So more local to region to understand why this is happening, is driving this kind of emissions. So I would like to thank you, all of you, for the attention. Thank you for the invitation to be here, sharing this with you. All our financial support. This is a big group. It's not just my work. It's led by Luciana Gatti. And our results between this time, 2010 and 2018, are free, available, the vertical profiles. So if anyone is interested, we could find in the DOI. And yeah, this is all. Thank you so much.
No, it's okay. I just had to turn it on. Thanks, Luciana, for this really great talk, and it's really great work, and it gives some excitement, I think, to people at ATTO, because before we all thought that the image from the GOSAT, we thought it was the Western Amazon that was more important. So now we know we're in the right region with the right footprint, so to really help with the methane cycle. So I was wondering, how much uncertainty do you think is in the emission ratios that you use for the fire estimation? And maybe it doesn't change your story, but is this something where, you know, continuous measurements like those at ATTO could help, or we know from the aerosols, for example, that we have a recent fire or older one?
Yeah, we know that we have uncertainty because we are using mean for these 90 years for all the dry seasons, and we have variability. When it's more hotter and drier, the ratio could be different. When we have over the years, this could be changing because we have climate condition changing already in the region. So for the estimates, we consider the variability between these 90 years in the duration to estimate the uncertainties. But of course, if you have much more another data sets that we could use to try to compare, because the ratios that we see from the vertical profiles are lower, so we have more methane could be emitting for less CO2, less CO than what we found in the literature. So these values are quite different, but we are talking about the measurements that is done until 4.5 kilometers high and not in the local near to the fire, so we could have this variability. But of course, if you have much more data to compare with this, it would be great.
Yeah. Well, my name is Jochen Schöngard. I work here in the wetland group MAUA of INPA. Thanks for this great talk. I have a question regarding the wetlands.
Yeah.
Now, the wetlands, we estimate they cover more or less 30% in the Amazon basin. But they are quite different in different types of wetlands. Now, most of them are flood pulsing. However, the flood pulses, they have amplitudes which vary quite a lot. So the largest amplitudes are in the southern region. In the northern basin, they are quite smaller. Here in the central part, it's about 10 meters, the annual flood pulse. And they also occur in different periods. Now, in the northern hemisphere, it's quite different compared to the southern hemisphere. And also, of course, they differ quite a lot in the vegetation cover. So you have, most of them are forested wetlands, and we know from these studies from Pangala that trees are channeling the methane formed under anaerobic soil conditions to the atmosphere. But we also have large complexes of quite open wetlands, like these white sand ecosystems, which cover huge regions in this northwestern part of the Amazon basin. So I was wondering now the next steps of your studies now to better qualify the emissions from the wetlands. What do you think might be next steps to get that ecological wetland footprint integrated in your data set?
Yeah, so the have much more information about the ecosystems and the habitats that we have in the wetlands, this could be really nice for we try to do a comparison, like a remote sensing comparison with our results. So now we are investing, Santiago it's working with me on this. We are trying to compare the bottom-up estimates from wetlands from the different ecosystems to what we are measuring in the vertical profiles to try to understand better in which regions we have these differences also. But like when we go, and one important point, when look at the northeast part that have these large emissions, our region of influence have influence from areas that is outside of Amazon basin hydrological area. So these don't have too much kind of wetland information. The Tapajós, not the Tapajós, sorry, the Marajó Island don't have too much kind of information of habitats, don't have a really nice map of wetlands. So this is a bit of lack of information for we try to understand better. So if you can have more information about these regions and what kind of ecosystems and how the process happens there, like with the isotopes signature to try to understand the process that is emitting from the trees and also from what kind of production we have in the soil, this could help. So my dream is have a kind of isotope analysis like in the Santarém region to try to understand better these large emissions and try to investigate better if this is coming from the wetlands, that's possible, and this is the main aspect that we think about. But yeah, we don't have too much information yet to try to complement this. Yeah.
Yes, hi, thanks for this really broad spatial overview on the methane fluxes. I was wondering if you are able to actually locate the spatial patterns between the wetland emissions and the fire emissions in terms of during the dry season that you have this obvious reduction in the wetland emissions and the increase, but are these actually co-located, so the occurrence of the fire is closely related to the reduction in wetland emissions, or if these are actually two completely separate things happening next to each other?
Okay, so the first point is that we are not completely able to distribute our emissions, especially because we are measuring one point that represents everything. So I cannot look like a remote sensing study that we see for each grid, how much is the contribution. Because we also don't have or didn't work with a proper footprint that we could estimate how much flux comes from each grid. But what we see, we relate with burned areas, the fire emissions, to try to investigate more or less what these come from. So we could try to relate from the concentration with the areas of burned areas, but we cannot properly point that this emission is coming from this region or other. So it's the same for biomass burning or the wetlands. We can try just to compare this spatial distribution, and with inverse modeling, we can do this better. We can distribute this like a spatial distribution, but with just the vertical profiles and the mass balance technique, we cannot point the spatial distribution. Then we cannot point if it's happening in the same areas or not.
Okay. Thank you for your presentation. I have one question related to this northeast part, that in the Marajó archipelago that you said, there's huge flooded areas there, and some of them are wetlands in grasslands areas. So there we have tons of cattle, especially buffaloes, in this wetland without land use. Mm-hmm. And I'm thinking how to separate the methane from the wetlands and from the buffaloes there.
Yeah, this is a good question. So we, like, for our kind of estimate, we are based on the other data sets, like EDGAR or national inventories that estimate enteric fermentation based on the number of cattle or buffaloes in the area and the emission factor. And then wetlands is just everything that is the rest. Okay? But if you have, like, isotope analysis, we could try to split this with the signature from the emissions from enteric fermentation and the wetlands, but this is not easy. It's not so simple. So this is a really big question. We are based, the anthropogenic emissions are mostly based on national inventories and emission factors that we have some studies that point how much come from. But from the vertical profiles, this is not possible. Just if you have some isotopes together with the measurement, we can try to understand a bit more if it's biogenic or if it's wetland or not. But just like we have the measurement of the concentration, we cannot point this. So we need to try to understand this with different methodologies.
Okay, thank you very much. And
Thank
you
you
again for coming to visit us. And I guess now, with great pleasure, we can invite Dr. Santiago, who will present, I guess, the work from his thesis, which he just defended last week. I think you got two. You can take a pick if they work. There's one on. I'll just give you this one.
Hello? Yes. Hello. Okay, let's see if this works. Yes. Okay. Okay, thanks. Yeah, so not so much introduction. I think I've been working here for the last 4 years. And I defended my thesis last week, so I will give you like an overview of what I presented there, some things that are general, not so technical, and some others that are a little bit more technical. So what you see here is the cover of my thesis, and what we wanted to show here as a message is that we can understand forest dynamics by looking at changes into the atmosphere or in some atmospheric components. So this is the topography around ATTO, and in blue you see the upland areas, and in green the low topographic areas. So the color scheme is inverted just to give this message of like we can understand forests by looking to the atmosphere. So it's a aquarela on silk screen. Okay, so why do we study methane and CO2? So Luana just gave a very nice talk about why we do this. But these are components of the atmosphere that represent a very low quantity of the air that we breathe. So it's about less than 1%, but they are powerful warming agents. And over the last 800,000 years, these gases oscillated between highs and lows every several thousand years. But today, I think these numbers are no longer up to date. What I saw on Luana's presentation is that we are already in 1,900 of methane, and we might be a little bit higher in CO2. So this is unequivocal evidence of the effect that we have had in the atmosphere. So this is quite a big argument on why we study this and why we look at this from ATTO, from our measurements. So methane and CO2, so they are produced anthropogenically and also naturally, and they circulate through the atmosphere, between the atmosphere and the surface. And they are important because, and the Amazon mainly plays a big role in the circulation because it is one of the largest carbon reservoirs on Earth, and it also, there has been some papers suggesting a decline in the sink of vegetation. And on methane, so we just had a very nice talk on methane, but this is also one of the tropics are one of the most uncertain components of the global methane budget. And the Amazon, only looking at the trees of the wetlands, they represent almost 4% of the global methane emissions. So we're talking about big numbers here. So this is why this is important. So I focused in my thesis. I focused on different spatial scales. So the methane study was more local, and then we went from like ATTO only studies to more regional, to a more regional perspective, integrating the ATTO data together with the aircraft data for CO2, but the same flights that Luana just mentioned. So I will start talking on the local study, the methane nighttime signals, and these were events in which the upper levels of the tower, or the top of the tower were showed concentrations that were higher than all the other levels. And this is unusual because the methane sources are at the surface, and doing further analysis, we found that they had like a seasonal pattern. And if you look to these plots, the radial axis on the left-hand side is the diurnal cycle, so this is showing the time, and then on the right-hand side, you see the months. So the red areas coincide, the red areas are the high methane events. So you see that the southeast part, so you have also wind direction there, and then the southeast part shows where, and the time and the month from where these events are coming from. So some of the first hypotheses that we have to explain the source of this methane was, or are at the moment, that they are coming from the river areas south of ATTO. And this means the river, the flooded areas, and also the valleys that you see here, so the baixios, as they call them here. So some of the hypotheses that we have is that, so we know, so the people, the ATTO community is quite aware of the dam that is built close to Porto Atto. So this dam produces methane. That methane can go downstream, so that's one of the things that have been researched before. But it also changed the flooding pattern of the river downstream, as it was shown by the studies in the group of Jochen, and this extended the flooding seasonality in this flooded forest, and this can lead to more methane, to more conditions that are methane prone, that can produce methane. And then it also killed a lot of trees downstream. So what we are doing now, and this I show this because I just want to advertise the posters that we have. We are looking into these sources with a more specific, with a different approach. So we're going to the valleys, we're going to the floodplains and the river, and we are measuring trees, water surfaces, soils, and this is some of the results. The preliminary results here are shown in two posters that are shown this afternoon, so check them out. What we want here is to try to get the isotopic signatures from these areas, and this is also connected to what Luana showed. Maybe we can understand this bimodal seasonality of the Santarém area, which might be helpful as well. If it's methane, let's say, if it's wetlands driving this. But this is a collaboration between the greenhouse gas group at IENA, the MAWA group, and also the forest dynamics group from Daniel Marra, that is somewhere here. So yeah, this is a big effort, and it would be great to have some discussions this afternoon. So now I want to shift to CO2. I will talk about what we did, the efforts understanding the CO2 signals at the upper level of the tower, and we mainly focus on simulating these CO2 concentrations using biospheric models and atmospheric transport models. So when we talk about concentrations, atmospheric concentrations, we talk about an integrated signal of many processes, also for CO2. Luana described it very well for methane. So we're mainly interested, or we looked at the net flux, which results in the concentrations that we see. But this is resulting from many different processes, net uptake, respiration, and all these things. But what we can see from the top-down approach is like the net flux. So this is the mean diurnal cycle of CO2 at the upper level of the walk-up tower. And what you can see here is that during the day, photosynthesis is active, and then the concentrations go down. We also have mixing and entrainment. So all these processes, like respiration, photosynthesis, and these atmospheric effects are driving these concentrations to go down. And then during the night, we have more stable conditions, we don't have photosynthesis, and then the concentrations go up because of respiration and also microbial decomposition. So if you look at this diurnal cycle, it has a very constant rhythm, but when we look at this at longer timescales, we can have a different picture, and we can try to get more information from this data set. So this is the monthly time series of CO2 at ATTO, upper level. And what you see is that there is an increasing trend, which is consistent with the global tendency. But you also see that there is quite some interannual variability. So years where CO2 is high, we can probably get some clues on why the vegetation did not take CO2 as it should. So we can have some clues on potential water stress or effect of high temperature or these things. So this is what I will show you after. So in this first approach, trying to simulate the concentrations at ATTO, we basically looked at the anomalies of the atmospheric data, which is the black line here, and the purple line is the anomalies in the flux data, so the eddy covariance flux. And some of the findings that we have here is that there are some years in which the anomalies in the flux data are decoupled from the atmospheric data, and this basically shows that at ATTO, the atmospheric signal can be completely decoupled from what we see at local scale in the fluxes. And then when we try to simulate the black line, so the concentrations, we use several biosphere models and an atmospheric transport model, and what we found is that, particularly for the response to El Niño in 2015 and 16, the models either anticipate this anomaly, as you see here, or they show a different sign. So what we suggest in our analysis is that there are different, so what is driving the source of error here, we suggest that is the biosphere models and not so much the transport, because there are some processes that are missing in these biosphere models, which is the response to drought, the seasonal leaf production that can be quite heterogeneous, as you see here in this image. So some trees can have no leaves, and then the tree just next to this one without leaves can be full with a full crown. So these processes are not in these models, and also the effect of respiration, the response of respiration to dry conditions. So in the regional study, we took the atmospheric CO2 record, so this one that I was just showing to you, and we integrate this with the aircraft data from INPE, and this is the network that we basically use in this study. There is ATTO there, all the sites that Luana mentioned before, and the darker areas are the areas that show a higher influence on our measurements, on the profiles and also ATTO. So you can see that close to the site, the colors are, the surface influence, it's a little bit higher, but we also have a spread out influence throughout the South America that is mainly concentrated to the equatorial regions and also outside of the legal Amazon boundaries. So if we look at the monthly time series of all these sites together, we see that there is, as expected, there is an increasing trend from low in 2010 in yellow to high, almost 410 ppm in 2018. The gray spots are just places where we couldn't have some data, so ATTO only started in 2013, like with continuous data, and then in 2015 and 16, there were some logistical problems with the aircraft data. But then this is what we have. It's not so many stations. If you think about Europe, for example, they have a smaller area, and they have more than 50 sites in the ICOS network, and we are trying to get net fluxes at a continental scale with 7 sites. So it's a little bit challenging. So I want to walk you through the inversion system that we are using to do this. So we have a set of prior information, so like the best guess of several flux components that they all integrated in the atmosphere result in the concentration that we see and that we measured at these sites. So for this, we need to have lateral boundary conditions. What Luana said as background concentrations is exactly the same. We also have anthropogenic emissions, fires, and ocean fluxes, and they are all coming from different products. And these are all fixed in our model, so we assume that we know this relatively well, and that we don't know, and there is more uncertainty in the biosphere fluxes. And from the biosphere fluxes, for the biosphere fluxes, we have 3 different models, so 2 process-based models and 1 diagnostic model that has different assumptions and different equations. So this is the part where we optimize with atmospheric data. So for this, we run forward, so we run all these models to simulate the concentrations at all the sites throughout the continent. We create simulated mole fractions of CO2, and we compare with the measurements, and we minimize these differences by adjusting the biosphere flux. And for each run with each of the biosphere models, we obtain a posterior flux, like an estimation of the fluxes after assimilating the atmospheric data. And then with this, we can get a mean posterior ensemble. And this is what I will be mentioning today, like the mean of all these runs with different biosphere models. So this is just to show you formally the priors that we use. So we have GFAS, EDGAR for fires, EDGAR for fossil fuels, and Ocean from the Jena product from Christian Rodenbeck. And also, I want to stress that what we do here is that we leave fires and fossil fuels, they are fixed, and then the net biome exchange is the one that we optimize, and when we combine fire, fossil fuels, and MBE, we get the total land flux. There is also one thing that we did in the ensemble, so we modified one of these priors, so it has no interannual variability and no seasonality, because we just wanted to see as a first guess, what happened if we give to the model a prior that is flat seasonally and also interannually, to see to what extent we get this information from the data set, from the atmospheric data. So you can imagine that if you want to know a little bit more about, let's say, gross like GPP or MPP, we would need to partition this MBE that is highlighted there, but that from atmospheric data is quite challenging. There are some ways to do it, but I think that with Carlos in one of the breakout sessions, we will talk about this more. Okay, so the first thing that we did, since we don't have a lot of measurements, we only have 7 sites, we tried to tackle the limitation by just looking at the regions that we constrain better. So how do we do is we have an assumed uncertainty before we do the assimilation, and then we have a posterior uncertainty. And in this plot, you see the colors match the colors in the regional map, and then there are some regions that have a high percentage, and that high percentage is the amount of uncertainty reduction after assimilating the data. You can see the Amazon region in this upper right-hand side. It has quite a high uncertainty reduction because most of the stations are inside the Amazon. But when also we look at different regions, like the Brazilian shield, which is number 3, it has also a high uncertainty reduction. And then number 8, which is the Guyanan shield, it also has a higher uncertainty reduction above 20%. And then the west of the Amazon region, number 1, It has like about 20% because the stations or the aircraft sites are a little bit farther east. But still, what we did here is that, okay, we don't constrain the entire continent, so we will just focus on the areas that have higher uncertainty reduction above 20%. So what we did is we basically kicked out all the places like the Central Andes, all the southern parts, the parts in the Orinoco region, in the savannas in Colombia and Venezuela, and the northern Andes. So all those places are places that we actually don't constrain with the data set that we have. So we focus mainly on these areas, and as you can see, we have the regions inside the Amazon, and also the Cerrado and the Caatinga. So what you see in this time series is the posterior ensemble mean, so all the models, so those are shown in bars, and the colors are matching the colors of the map. And the markers, the triangular markers, is the posterior, so that's before assimilating the data. So you see, the first thing that you see is that there is a change, quite a big change in the Cerrado and the Caatinga. So there is a decreasing zinc in the Cerrado, and the Caatinga actually changes the sign. You also see, I don't know if I have a pointer. No. Okay. So when you compare also the main branch of the Amazon River, when you look at the dark green, which is the first bar, we see that there is a decreasing zinc as well, and it's very close to neutral, which is also quite comparable to what was published in the SPA, in the science panel for the Amazon, where they say that the rivers are almost neutral. And then looking at the other regions within the Amazon, when you compare the Guyanan Shield with the southern part, the southern part shows like a larger sink of carbon, but then in 2015 and 2016, this is reversed, and this might be because of the effect of ENSO or El Niño. So we have some regional differences, as you can see here, but let's look at that with more detail for the regions in the Amazon. So this is the seasonality that we see in the regions inside the Amazon. You can see that there are 3 lines that are very close to each other, showing quite the same shape of the seasonal cycle. And then the Brazilian Shield, which is the southern part, has a very different seasonality. And then this is the seasonality after assimilating the atmospheric data. So we have different peaks, we have a different shape of the mean seasonal cycle. This is considering all the stations, ATTO stations and aircraft stations. And when we take ATTO out, the region that suffers or that changes the most is the region of the Guyanan Shield, which is mainly where the footprint of ATTO is. So we see that at this scale, having ATTO in the assimilation framework has quite an import, or plays a big role here. And I will show you more of this in the next slides. So spatially, we also can resolve these fluxes spatially. And in green, you see the carbon uptake, and in purple, the carbon release. So here, I just want to show you some ideas of what is the variability in space of the carbon fluxes. In 2010, there was a source in the western part of the Amazon that we don't see in 2014, but we do see in 2014 an enhanced sink in the southeast part of the Amazon. And then comparing 15 and 16, the source in the eastern part of the continent seems to be stronger in 2016. So these are some features of internal variability, but also you see that there is a consistent sink outside of the Amazon in the semi-arid regions of the Caatinga and the Cerrado, which we are still investigating. And looking at these fluxes, when we take each individual model and we look at the closest grid cell, the closest to ATTO, you see all these colored lines that they are very different from each other, yeah? So they are all over the place. And the observations from ecovariance is the black line. When we assimilate the atmospheric data, we get this plot, so all the models are closer to each other, but they are also closer to the black line, so the observations. But when we take ATTO out, you see the dotted gray line, which is totally anti-correlated with what we see with the ecovariance data. So this is another argument of the potential of having ATTO to constrain net fluxes, as I showed regionally and also at a local scale compared to independent data like ecovariance. So this data was not used in the model, it's completely independent. Okay, so with this, I think just on time, I will leave this just final remarks. And if you have questions, I'm happy to answer them. Yes.
Santiago, beautiful, really nice job. On your map of the carbon source and carbon sink, you showed 3-4 slides ago, you stopped the analysis in 2016. And at that time, you know, all the regions shows as a carbon, yeah, this one. So shows as a carbon uptake. But the recent work of Luis Aragão and many others shows that even the Western Amazonia could recently becoming a carbon source. So the question is, your map does not show that for the Western Amazonia. So this is because you stopped in 2016 and this process could be more recent, or the process are not fully represented on your models.
Yeah, so this is a selection of all the maps. We do have the same for 11, 12, 17, and 18. But you're right, we don't see in the western part of Amazonia, we don't see, we actually, in the time series, in the bar plot that I show, the western side of the Amazon had the highest uptake. And I think that from all the regions that I selected to do the analysis, this is the region that has the least or the smallest uncertainty reduction. So from 2010 to 2012, the aircraft profile in the west was in Tabatinga, so that was like 5 degrees, a little bit more to the west. And then after starting in 2013, it moved to Tefe, and then you can see in the uncertainty reduction plot that this affects this particular region. So I think that with the data that we have, this is the region that we constrain the least. And another thing is that this is just an ensemble of 3 different biosphere models. And when you look at the prior shows a really big sink everywhere. So just getting these spatial differences from year to year, I think it's already an input of the atmospheric data. But we do, yeah, we acknowledge that we can have some biases just because the ensemble is too small, I think. So one of the things we want to do is to do more tests with other models, or maybe a statistical production of models that we can perturb these fields and then do many inversions so the ensemble is larger. So this is ongoing process, yeah.
Hi, Santiago. Nice talk, thanks. I was a little, I guess I'm a little confused about the difference between sort of what you did here and what Luana talked about. So for her product, she was subtracting the fire using the emission ratios from the methane and then modeling that, right? And what you're doing is you're including in the prior the GFED fire emissions as a part of your prior, right?
Yes.
So I'm just wondering if you might get different answers if you try to subtract the fire from the atmospheric record and then leave it out of your bottom up, because part of the interannual difference is going to have to be the fire.
Yeah, so
emissions.
So the first thing, the easy answer is that we know that the fires that we use here are low. Because GFAS, so there was a study recently that they showed that only GFAS in high fire years can be a factor 2 lower. So we acknowledge that this product is low in magnitude, but the way in which the inversion works is that you need to give the model all the prior fluxes that you know they contribute to the observation. If you don't, then your posterior will contain that information because you are seeing everything. But then partitioning that is the challenge. So I think that from Luana's approach is different because they get first the fire influence, and then they subtract that from the observations, and then that's, let's say, like the without fire component. We give here to the model everything, and then we get a net flux. So it's just a different approach.
No, I was just wondering if you could use that same approach for the CO2 inversions.
Yes, we could, but yeah, I have to, yeah.
Yes, nice presentation. I have a question related to the diurnal cycle of the CO2 fluxes, or where you showed the CO2 concentration and the respiration part versus the photosynthesis part, and also this is somehow related to the results that you were showing about the methane concentrations, where you saw that the sources of the methane are actually located at the upper level of the tower, while you have lower concentrations of the methane close to the surface. And I was wondering, from the respiration point of view, you would assume that from the vertical profile at the tower you see higher concentrations close to the surface as well, right? And lower concentrations at, let's say, the top level. Can you confirm that for the CO2 concentrations?
So this is the top of the tower.
Ah, this is all top of the tower.
This is only top of the tower.
Okay,
Yeah.
And then I was wondering that the strong reduction that you see on that slide during starting with the sunrise, basically, if that is also to a certain degree due to the growth of the planetary boundary layer.
Yeah, right.
So that if you have a source at the ground and you have lower concentrations actually in the higher parts or above the planetary boundary layer in the early morning, you have the mixing in of lower concentrations, that this is actually causing also a part of the strong reduction.
Yeah, so that, I think you cannot get this reduction without entrainment.
Yes, exactly.
So there is a component of entrainment, which is, I think, big, and there is a component of uptake of vegetation.
And did you, or did someone ever look into this, like how big the entrainment component is with respect to the photosynthesis?
No, that I know, but it's an interesting question because, yeah, maybe the meteorologists would say that the entrainment is bigger, and the ecologists maybe say the uptake is larger, but I don't know. I have no idea.
Yeah, and then associated to that, if you see the high CO2 concentrations at the top level, I mean, how does this correlate with the methane concentrations, where you also have the source from the ground, but you have somehow an inversion of the profile that you would expect there?
Yes, I have. When you look at the diurnal cycle of both methane and and CO2, and also actually radon, they are very similar. So there is this flashing peak in the morning, and then this drawdown in the day, and then the buildup in the afternoon. But what is different for the methane diurnal cycle is that during the dry season, there is not this increasing part after midnight, because sometimes it's already high, you know. And I think that one of the issues at ATTO is that we don't really know what's if this level, which is 80 meters, is inside the nocturnal boundary layer, or if we are in the residual layer. But for the methane study, we kind of assumed that we were in the nocturnal boundary layer, because the differences were so high that we assumed that this has to be a source, a local source, somewhere else. But then the question is that what if we are in the residual layer? Then it's like concentrations of the day before, right? Like that would be more connected to so it's a potential way forward, I guess, because there are still some open questions in that study.
Thank you.
Okay, unless there's any pressing questions, I guess we're into the coffee break already. So,
just a comment on the entrainment question. You can actually see the entrainment rate. We've done this for heat flux, for the entrainment, heat flux entrainment in the early period of the boundary layer growth in the tower, while the boundary layer is growing and still hasn't reached the top of the tower. It's very nice. The heat flux entrainment shows very nicely. I'm sure it will be the same for CO2. Then you have quantitatively precise measurement.
Nice.
That's a very useful comment. I'll come back to you later. Okay, thank you, Santiago, and congratulations on being a doctor.
Okay, today we were so eager to get into the science that we skipped some things. So I just forgot to give an overview of the program, but I hope that most of you have read the program already. So what we have now is coffee break. I think we are all happy about that. And then we will continue with the online sessions. So it's actually the people that could not be present but wanted to submit a poster that they will present online. Then we will have a small recognition of the people, of the local ATTO staff. And then we will have lunch, and then we will continue with the poster sessions. Please, for the people who are presenting the posters in person, just check with the poster organizer, because they will be collecting your presentations if you have one, or they will just check with you how it will be done. So please, yeah, we are all happy now to go into the coffee break. Okay. So we have now the poster sessions. We start with the poster session. So we have the online posters that have been submitted. And we have the presentations from the persons that sent that. But they are also connected online. So at the end of the sessions, when all the posters have been presented, we have the chance to ask questions to the online authors. And the posters also are going to be visible in Gather Town. I sent you the link, but I will be happy to send it again in case you don't have it. Yeah? So all posters, not only the ones online, but from the whole presentation, if you have submitted or if you have uploaded it, they should be available on Gather Town. Okay, so we start then with the online poster session. And the first poster that we have is the one from Carla Souza, from the Instituto Federal de Educação, Ciência e Tecnologia do Pará. And the title is Climatologia da altura da camada limite atmosférica na Amazônia Central. So please, the first video. And I hope that Carla is also connected online. I think we have probably to wait a little bit of a moment for the Zoom page. So, okay.
Atmosférica na Amazônia Central. O nosso trabalho ele fala sobre a altura da camada planetária na região da Amazônia Central. O diferencial dessa pesquisa é que a gente apresenta uma climatologia para essa região com um quantitativo de dados bem robusto. Nós temos 6 anos analisados, de 2014 a 2020.
Pode empezar outra vez o vídeo, por favor?
Como metodologia, a gente usou a climatologia da altura da camada limite atmosférica na Amazônia Central. O nosso trabalho ele fala sobre a altura da camada limite planetária na região da Amazônia Central. O diferencial dessa pesquisa é que a gente apresenta uma climatologia da altura da camada limite atmosférica na Amazônia Central. O nosso trabalho ele fala sobre a altura da camada limite planetária na região da Amazônia Central. O diferencial dessa pesquisa é que a gente apresenta uma climatologia para essa região com um quantitativo de dados bem robusto. Nós temos 6 anos analisados, de 2014 a 2020, com exceção de 2017. Como metodologia, a gente usou um ceilômetro instalado no sítio ATTO, localizado na Amazônia Central, para determinar a altura da camada limite planetária. Foram feitas medidas de 5 em 5 minutos. Diante disso, nós conseguimos os seguintes resultados. Anualmente, observamos que a camada limite planetária atinge o seu pico por volta de 20 horas UTC, aproximadamente 1.500 metros, e conseguimos observar nesses anos que 2015 ele apresentou o maior pico, isso está associado ao fenômeno de El Niño que aconteceu nesse ano, assim como nós vemos que no ano de 2016 a camada limite apresenta o menor pico, e isso está associado ao fenômeno de La Niña nesse ano. Fizemos também um comparativo entre a estação seca e a estação chuvosa e podemos observar que na estação chuvosa a camada limite tem menor altura, que está associada a maior cobertura de nuvens durante esse período, que dificulta a radiação solar incidente na superfície e, consequentemente, menores turbilhões energéticos que vai acarretar numa camada limite planetária menor. Comparamos também a camada limite nos anos de El Niño de 2015 e o ano de La Niña de 2020 e vimos que 2015 teve uma maior altura, isso está associado a menor cobertura de nuvens que acontece durante esse fenômeno, que facilita a radiação solar e, consequentemente, maiores turbilhões e maior camada limite planetária. A gente consegue observar que o mês que a camada limite atmosférica vai ter uma maior taxa de crescimento, assim como maiores alturas, vai ser setembro, que está associado à estação seca dessa região. Assim como nós vimos que as menores taxas e as menores alturas está referida ao mês de maio, que também está associada à estação chuvosa dessa região. Então, a gente consegue concluir que a altura da camada limite planetária apresenta sazonalidade, influência direta de fatores diversos relacionados ao tempo e clima na região da Amazônia Central.
Hello everyone, my name is José Mantovani. I'm a research associate and PhD student at IMP. Today we will talk about part of my master dissertation entitled Assessing the Performance of PBL Parameterization Schemes in the Central Amazon Basin during Go Amazon. The PBL schemes are responsible for parameterizing vertical subgrid scale turbulence in numerical weather prediction models. The performance of these schemes depends on meteorological conditions as well as geographical features, which makes validation and testing a necessary procedure to get confidence in their applicability. However, few studies evaluated their performance in the Amazon rainforest so far. In this context, 11 PBL schemes were tested through short-term forecasts performed within WRF model version 4. The results from the 1 km resolution domain were evaluated with in-situ observations collected at the T3 site. The model domain configuration is shown in figure 1. The PBL-H time series revealed that PBL schemes in general roughly captured the daily cycle present the best agreement during daytime under dry conditions, according in figure 2. The observed PBL-H is underestimated in both conditions. The error statistics for predicted PBL-H were computed using the continuous allometer estimated PBL-H for daytime and nighttime, which indicates that diurnal PBL-H is better reproduced in comparison to nocturnal. The nocturnal PBL-H is better reproduced in the wet period. This may be related to the very stable conditions observed in the dry period, which explain the poor performance at night for almost all PBL schemes, except the local Mallory Yamada Nakanishi-Nino level 2.5 that outperforms all others. Through figure 4, the PBL schemes showed a good agreement with vertical structures during the wet period. In contrast, they showed difficulties in forecasting the profiles under dry conditions, especially in the morning. The PBL schemes tend to predict warmer and drier conditions within PBL, mainly non-local schemes. The PBL-H spatial fields in figure 5 revealed how PBL schemes distribute the PBL-H across the basin. For instance, non-local PBL schemes produce much more pronounced land-river contrast than local schemes. The hybrid quasi-normal scale elimination scheme produces the deepest PBL-H, and the University of Washington scheme the shallowest ones. The results revealed that non-local scalar Shinhong scheme is the best option for the PBL-H prediction in the wet period, while the local Mallory Yamada Nakanishi-Nino level 2.5 is the best for the dry period. The error statistics computed for surface meteorological variables and turbulent fluxes, not shown here, suggest that the dry period is overall better predicted. Thank you for watching and feel free to contact me. Bye!
Okay, I think we have a problem with the audio. We can see if maybe Flavio is connected online and he can present his poster online directly. So we go to the next poster, and that's from the greenhouse gases. That's a difficult name. Him, University of Wageningen. The next poster.
Hello, welcome to this pitch. My name is Wout Blankstein and I'm happy to present you the results of my thesis. It concerns the application of the radon trace method to infer methane fluxes at the ATTO region. We wanted to try to reduce the current uncertainty in regional natural greenhouse gas nighttime fluxes. In the early 1980s, the radon tracer method was proposed as a powerful tool to determine those fluxes. And as we have current radon concentration measurements at the ATTO site, it was possible for us as first time to apply this radon trace method to obtain methane fluxes in the region. I want to start by introducing you to this unique radon concentration dataset. You can see here in this figure that the wet season concentrations are much lower than the dry season concentrations, and that is caused by a change in soil moisture and a shift in the nocturnal boundary layer. Those radon concentrations are used together with the change of greenhouse gas concentrations to obtain a methane flux, and for that we use as input a radon flux. However, this radon flux was not yet defined in the region, and therefore the first part of the research consisted of calculating a radon flux, which we could use in the second part of the research to calculate a methane flux. The results of the radon flux calculation were rather good. The radon flux, which we calculated at 0.017 millibecquerel per square meter per second, cohered with chamber measurements that were performed at the ATTO site before. What we also found out is that there are two different methods to come to this flux, and that this method was essential for the final flux result. We use the radon flux as an input to the methane flux calculation, and here I want to show you the methane flux result, and we see that in the dry season we obtained a rather high methane flux compared to the wet season. This is first an intuitive result, as you would think that in the wet season there are more anoxic circumstances, resulting in more methane emissions. However, it was a quite case-specific result, as we found out that it had to do with the change of wind direction. In the wet season, the wind came mainly from the east, and the east are the upland areas in the region, and those upland areas are known for a rather low methane flux. However, in the dry season, the wind brought more air from areas with a higher methane flux, as the wind came more from the river areas. What I want to conclude with is that the radon tracer method could be well applied to infer methane fluxes, and especially at the ATTO site. And the methane fluxes that we present did well cohere with other studies that showed quite similar methane flux.
Okay, the next poster is from Jordi Villa from the University of Wageningen about the Cloud Roots campaign.
Hi everybody, bom dia, buenas tardes. My name is Jordi Villan, and I would like to present in very short the first results of the Cloud Root campaign that took in August 2022, approximately 2 months ago. And our aiming is to advance the understanding of in-canopy biochemical and physical processes at clouds in the Amazonian rainforest. The overarching question is that to reduce a bit two of the largest uncertainties that we have now in climate models. One is the CO2 exchanges and the other is cloud formation. To do this, we have a methodology that is very rich. We go from very comprehensive observations, that's the one that I will present in 1 minute, to high resolution or largely simulation. I'm talking about 50 by 50 by 20. So the main thing is that we have certain uncertainties. This is a typical map of the Amazonia with large uncertainties in the future of cloud cover up to 2.7. And what we see is that clouds are influencing very much the radiation, and as a result, photosynthesis is going to change. At the bottom, we see that photosynthesis is acting. These are very preliminary results collected 2 months ago. And we see that in the morning, photosynthesis is very active with small conductivity grades of the leaf. But in the afternoon, we have a midday depression. These results will be translated to the larger simulation to understand how is the ejections and sweeps of carbon dioxide, water vapor, and sensible heat flux. And afterwards also we have a lot of soundings to know how is the thermodynamic states of clouds. In very short, if you want more details, just contact me, and I can tell you more about the cloud routes and the framework. Thank you very much.
The next poster is from Gilmar Enrique Santana da Silva, del Instituto Federal de Educação, Ciência e Tecnologia do Pará.
Olá a todos, meu nome é Gilmar Henrique, sou aluno do Instituto Federal do Pará, campus Belém. Sou graduando em licenciatura em Física. Estarei apresentando o trabalho intitulado como Linhas de Estabilidade na Região Amazônica e o Transporte de Gases próximo à superfície, durante a ocorrência de downdraft. Bem, neste trabalho foram observados que essas linhas de estabilidade, que são geradas ao longo do norte da costa do Brasil, e seus efeitos sobre a sua estrutura atmosférica durante seu deslocamento para o oeste da Amazônia. Bem, vários estudos têm mostrado que essas linhas de estabilidade podem causar intensas correntes descendentes de ar na região amazônica, conhecidos como downdraft. Imagens de satélites foram usadas para a identificação dessa ocorrência de LIs acima da Amazônia Central. Bem, alguns resultados que serão mostrados aqui serão baseados em comportamento do CO2 e do ozônio. Como a gente pode observar na figura 1, o ciclo diário médio do CO2 para o ano de 2014, acima do sítio ATTO. A linha tracejada corresponde ao período seco, de julho a dezembro, e a linha contínua corresponde ao período chuvoso, de janeiro a julho. Como vocês podem observar, CO2 a gente tem tanto para o ano de 2014 quanto para o ano de 2015. No ano de 2015 se observa que a concentração do CO2 se encontra abaixo de 400 ppm, enquanto que em 2015 são acima de 400 ppm. Para o ozônio, a gente pode observar, tanto para o período chuvoso quanto para o período seco, que no período chuvoso não aconteceu nenhuma alteração considerada entre o período chuvoso entre os 2 anos, 2014 e 2015. No entanto, para o período seco, se teve uma leve alteração nos seus valores de concentração durante os dias. Aqui a gente tem o comportamento a variância do ozônio, a variância média do O3 durante a ocorrência de linha de estabilidade acima do ATTO. O time 2 indica o horário da passagem da linha, onde a gente pode observar o que aconteceu antes da passagem dessa linha e depois da passagem da linha. Durante a passagem da linha a gente tem uma grande variação na concentração desse ozônio. A mesma coisa acontece para o ano de 2015. Já para o CO2, a gente tem dados, não se houve dados suficientes para análise, para se obter a variância do CO2. No entanto, a gente pegou dias do CO2, onde se houve essa alteração durante a passagem da linha de estabilidade. Como a gente pode ver, na concentração do gás CO2, durante o dia 18 de janeiro de 2014, a LI esteve acima do ATTO, às 11 horas. Já na figura 8, a concentração do CO2, durante o dia 16 de 4 de 2015,
esteve acima do ato às 11h30. Bem, essa é uma breve apresentação sobre o meu trabalho. É isso.
Good. The next presentation. Yeah, the next presentation is going to be from Nora Zanoni from the Max Planck Institute for Chemistry about the atmospheric.
Hi everyone, I'm Nora Zanoni and I'm presenting a poster about the atmospheric impact of sesquiterpenes emitted from the Amazon rainforest. So why are we interested in sesquiterpenes? This is because they're highly reactive biogenic VOCs, and despite being highly reactive, we measured substantial concentrations of these molecules in samples taken from the ATTO tower, including at 325 meters, which means that there must be strong sources of emissions of these compounds in the forest. We also noticed remarkably different composition when samples were taken from the tall tower or next to the understory. Also, previous works in the Amazon rainforest reported significant emissions of sesquiterpenes from soil, cryptogams, and the leaves of one hyperdominant tree species. So we wanted to know better how tree leaves contribute to the emissions of sesquiterpenes in the Amazon forest and what is the ultimate atmospheric impact of these compounds. Especially, we wanted to know the contribution in terms of what type of compounds and concentration. So to determine this, we conducted a field experiment during December 2021 by using some plant chambers that were installed at the instant tower. So we chose a healthy branch from an individual of a tree species that was already identified and measured for other properties and studied for other properties next to the instant tower, so that was accessible from there. But also this tree species is a dominant one in central Amazonia. So we use PTRMS and GC-TOF-MS to measure the composition of biogenic VOCs and specifically sesquiterpenes. So in the poster, I'm showing some preliminary results. I invite you to have a look at the poster. So I use isoprene as a reference compound because we know how this varies with light and temperature. You can see the concentration of total sesquiterpenes measured by PTRMS in the B and D graphs. So to determine if these emissions are depending on light and temperature, we need to do more work. But what I wanted to show as well is that just after we installed all the chambers and we enclosed the branch, a strong storm happened at the ATTO site, and this strong storm caused some damage on some leaves that were enclosed in the chamber. So when the storm finished, we removed these leaves. But what happened is that as soon as the damage occurred, we saw a spike in the emissions of sesquiterpenes. You can see it here. So these emissions then leveled down with time. But we can see clearly that the sesquiterpenes emitted from, so we can see a sample of the damaged branch and a sample of the healthy branch. We can see that in terms of quantity of species, they look very different. We had 1 order of magnitude difference in concentration, but also in terms of diversity, they were very different. Here you see many more compounds, many more sesquiterpenes emitted when the branch was damaged and very few ones when the branch was healthy. But what we noticed that in both the healthy and the damaged branch samples, these three species emitted sesquiterpenes first, and it emitted one compound, alpha-copain, that was the one with the largest concentration that we measured in the samples taken from the ATTO tower. So this was very interesting for us because it means that these three species is one source of emission of sesquiterpenes in the forest. So if you have, of course, we need to do more work on the data, so this is very preliminary, but if you have any suggestion, question, or doubt, do not hesitate to contact me at the meeting. I will be attending online or by email. Thanks a lot.
Next poster, also from the Max Planck Institute for Chemistry, Giovanni Pugliesi, also on aerosols.
Pugliese, and I am a postdoc in the Jonathan Williams group at the Max Planck Institute for Chemistry in Mind. And today I'm going to show you the preliminary results of our experiment conducted at the ATTO site, where we measured the fluxes of the volatile organic compounds from the forest soil. This is because the soil may significantly impact the atmospheric VOC concentration, as it can both emit and consume atmospheric VOC. So in this study, we continuously monitored the VOC fluxes from the Amazon soil over 10 days. And the soil VOC fluxes were measured by means of PTR-TOF-MS, which was directly connected to an automated soil flux system consisting of a multiplexer, a gas analyzer for soil respiration measurement, and 4 long-term automated chambers. 3 of the 4 chambers were placed on 3 different types of soil. One was a bare soil with a high organic content, the second was a bare clay soil, and the third one was the clay soil covered with a litter. The 4th chamber was placed on a Teflon and was used as a blank to take into account the background emission from the system itself. The chambers were measured consecutively, and the fluxes measured from the blank chamber were subtracted from the fluxes measured from the soil chambers. In this slide, I'm showing you the results for isopene, which was taken up by all 3 types of soil, and the isopene soil uptake also showed a Diel cycle, which was most probably due to the Diel cycle of its atmospheric concentration. The highest isopene uptake was observed for the organic soil, suggesting a higher microbial activity in this soil compared to the other 2 types of soil. This was further confirmed by the soil respiration, which is a strong indicator of the microbial activity within the soil. Indeed, highest soil respiration was observed for the organic soil, and it was even up to 35 times higher than the soil respiration observed for the clay soil. In contrast to the isoprene, monoterpenes and sulfur-containing compounds like dimethyl sulfide and metanthiol were emitted by all 3 types of soil, and especially for monoterpenes and metanthiol, the highest emission rates were observed from the soil with the litter, indicating a higher production of these compounds from the litter degradation. In conclusion, the soil VOC fluxes, but also the soil respiration, were significantly different between the 3 types of the soil that we investigated, indicating an heterogeneity of the fluxes from the Amazon soil. In future experiment, we will first further optimize our analytical setup, especially to reduce the background emission of some VOC, and then we will deploy the system for a long-term measurement in order to investigate the annual and the seasonal variation of the VOC fluxes from the Amazon soil. If you want to know more about our experiment and about our results, please come to see my poster at the end of this session. Thank you very much.
Okay, next presentation is from Bettina Weber from the University of France.
Dear ATTO community, I'm really sorry that I cannot be with you during the workshop. Indeed, I would have loved to do so, but I'm glad that at least I can give this short presentation. We just got funding, and I want to give you an idea about our research plans of the next years. The topic of our research will be on biosphere-atmosphere interactions of cryptogamic communities at ATTO and their relevance across spatial scales. As most of you probably realize, there is a wealth of fungi, lichens and bryophytes at ATTO, and we want to learn more about their relevance in different processes. First of all, we plan to map the cryptogam communities at ATTO. For this, we will assess imagery with a drone, and then we will classify the major groups of cryptogam communities by utilizing image segmentation and also artificial intelligence. As an output, we will learn about the relevance of cryptograms in general and also of the different organism groups. And this will then also facilitate an upscaling of processes to ecosystem scale. In the next task, we want to continue our microclimate measurements, and we also want to expand these. Some of you probably know that we conducted microclimate measurements for quite some years already on one tree, and there we measured at different heights. But one tree doesn't tell too much because we also have to consider really variability at different sites, and for that we want to expand these measurements. So the plan is to conduct long-term measurements at 3 different heights and on 4 trees each in the Campina and the terra firma forest. We will also have an automatic data transfer to the server. This is, at least is the plan. As an output, we will learn about the activity patterns of bryophytes. We will also find out about the environmental small-scale conditions, and we hopefully also learn more about the relevance of bryophytes in water cycling. In the third task, we want to investigate the CO2 fixation and carbon cycling in bryophyte communities. For this, we will conduct CO2 gas exchange measurements, both under natural conditions in the field and under controlled conditions in the lab, and we will combine these with the microclimate data in order to come up with a carbon balance of the bryophytes. These data will give us also hints on the carbon storage capacity of bryophytes, which will be very interesting, as we think. Next, we want to continue our measurements on bioaerosols. This is in close collaboration with Christopher's group. So we will continue the bioaerosol emission measurements by means of different techniques, and we will link these data also to mapping data that we have. As an output, we want to learn more about the relevance of cryptogram communities and also of the different organism groups as a bioaerosol source. We want to learn more about the relevance under varying environmental conditions, and this should also serve as a baseline to evaluate the relevance of bioaerosols in climate processes. Finally, we also want to continue our studies together with a group of Jonathan Williams. Here, especially Achim Edbauer was working on these things, and we studied the emission of VOCs. In previous studies, we found out that especially bryophytes emit quite a lot of VOCs, and especially sesquiterpenes, and these emissions are of similar magnitude as those calculated for the canopy. So the organisms indeed seem to be relevant, and this is why we would like to continue and intensify these measurements. So our future plan is to investigate the environmental conditions under which VOCs are emitted, and to increase also the number of investigated species. As an output, we expect to get the parameters that are relevant for VOCs emissions, and these will hopefully give also hints on the underlying mechanisms. And with these data, we can also confine the overall emission values. So this is already all what I wanted to show. Thank you very much for watching and listening. In case you have questions or you have ideas for collaboration, please contact me, or also talk to Sibeli Barbosa, who is at the workshop. Thank you very much and have fun. Bye-bye.
Welcome to the short presentation of my poster, which has the title Using BEXIS2 as an efficient research data management system for the ATTO research project.
My name is Marcus, and I'm responsible for the data management of the ATTO project. From the data management point of view, the main challenge is to provide a functional platform for consortium internal and external transparency, traceability, and data exchange between a large number of institutions and research groups that are involved in the project in order to foster collaboration and maximize the output of the project. We use Bex's tool flexible and modular data management software to provide efficient data management and exchange within such a large research consortium, and to allow long-term data archiving, documentation, secondary analysis, data reuse, and accessibility for the scientific community. Thus, we have the possibility to organize the scientific data life cycle very efficiently. Due to a comprehensive documentation for more than 80 metadata entries based on FAIR principles, and sharing and publishing of scientific data through the possibility of assigning a digital object identifier. This meets the requirements of the funders and the requirements of the journals to share scientific data. The DOI structure is based on the version number of the respective dataset. Currently, we do have 25 datasets with a registered DOI, and the download numbers, which are more than 550 downloads just of these 25 datasets, show that there is a huge interest in our datasets. We do have 126 uploaded highly diverse datasets from various scientific branches, starting from meteorology to soil science. An overview of which datasets have been collected at which location and are available can be seen in the second part of the poster. In conclusion, we can summarize that we have an effective tool which has an increasing number of users in recent years and which facilitates an exchange of datasets within the consortium as well as externally. And finally, I would like to encourage you to send me any needs or ideas of tools, features, and wishes for the ATTO data portal to be implemented since it will be developed further in the future. Thank you.
Okay? Okay. Oh. One moment. Flavio, you can start with your presentation.
Everyone is seeing my screen. So. Can I start or?
Okay.
Okay, so my name is Flavio de Oliveira. I'm here to present my works called The Effect of River Breeze on Gas Transport in Central Amazon. So this is a work as a part of my PhD thesis. So for this work, we ran simulations with WorthCam for the first 5 days of August of 2014, that is the dry season in the Amazon, to see the influences of river breeze on gas transport. So we decided to see two different regions in the river breeze in the contrast with the forest region that is represented by the square number 1, and in the urban region is represented by the square number 2. So this is represented by the land use that is represented in the WorthCam. And then we done with the comparison between wind direction and speeds and the gas concentrations in the 3 different sites for the Go Amazon sites as well. So for the first results, we have looked into the temperature difference and 2 meters in the forest region and on the urban region. And we've done this every day in 15 UTC, because when we observe the river breeze, that is the same time that all of the days that we can observe the river breeze is present at this time. So we can observe for the river breeze was present in the 2 days, that is the 3rd and 4th of August, even for the forest region and the urban region. And when we realized that the vertical cross-section from the gas in the day 4th of August, that it was the river breeze more intense day, we can see that the river breeze was responsible for trap highest concentrations of carbon monoxide that is showing on this figure, and for ozone as well. And the same, we can observe this on this red rectangle, was observed for the urban region that is represented by these 2 dashed lines on this image. And that's the river breeze was responsible for trap highest concentrations of carbon monoxide and the ozone on the west region of the city of Manaus. That is a region of mainly habitated for populations. It's not for industrial region. And this is just a resume of our work, and I invite you to visit the poster in the end of these presentations, and feel free to ask questions. Thank you.
Okay, thank you, Flavio. So we have all the poster authors connected online in Zoom, so if you have a question, please, you are free to place your question now. Any of people that presented the poster. Any questions? Okay. Sorry, as you see, it's a little bit to communicate with the people behind the window that is managing the technique here. I guess we should have learned some sign language before that. So if there are no questions, but please feel free to contact the authors from the posters. They are online connected now, but you have the addresses, and as I said, all posters have been uploaded into Gather Town, the same platform that was used last year for the online workshop. Okay, then I guess we continue with the program. And the next part is this, we wanted the people that work at ATTO, the main staff, and we have okay, we have first the presentation from Jürgen, but I don't know if we should move the presentation from Finlandia before. Yeah? Okay. Você pode.
I think I have to share my screen.
Okay, so we go ahead first with the presentation from Jürgen. So, Jürgen, I think you know Jürgen. She's from the specialized in atmospheric biosphere interactions, biogenic traces, open. Now he's retired and neuroscientist from the
Viviana, I don't know whether you can hear me. The screen is fine.
Yes, we can hear you.
Okay, so
Just
I can
wanted
just
to.
start.
Okay.
Perfect. So we learned a lot during this pandemic, doing all these virtual things, right? Yeah. Well, I regret not to be in Manaus. I booked all my flights and the hotel already, but then I had to cancel it for some health reasons, and I had to stay here in Mainz. But I think, anyway, what I want to say is thank you to a lot of people who accompanied my active time at ATTO, and I would like to address especially those people who are working in the background. And just to start with, for those people who are not so familiar with the history, well, these were the people where the ATTO virus was born and spread. Andy Andre was the one who was so heavily involved with all this tropical research. He infected me, and we had our connections to Manaus, to INPA, and so I came together with Antonio Manzi, and we discussed a lot about our things. And well, so ATTO developed more and more, and the INPA director at that time, Alberto Luis Val, he also was enthusiastic, and we were lucky to have two politicians just to get into this game. And one was Frida Meyer Kramer, and the other one was Luis Antonio Barreto di Castro from the MCTI. And just to give you a feeling about this, what happened, it was in 2007, so this was the start of this idea of ATTO within Brazil, when the State Secretary from the BMBF visited Manaus, visited INPA, and wanted to see what we are doing there. So we went out there and we had some discussions, and he asked what we could do and would like to do. And so we just discussed this idea of building a very tall tower. And then during the next years, and you see this were a lot of years, 2014, Antonio Manzi and me were standing on the tower, which we are in the building phase, and 2017, the tower was more or less ready, and meanwhile, he's fully equipped with a lot of instruments, I learned, and it's really great. So it took years, and within these years, there were a lot of steps from the memorandum of understanding on the political side to be signed, from the project description and submission and the grant of the project, up to a lot of things from the access road. So you do not need to read this. I just want to demonstrate it took us full 10 years to get everything running at the end and to get it into a shape where it could be used for scientific research. And during this time, there were a lot of people working with us and supporting us, and some of them always stayed in the background, and I would really like to mention these people by name. I'm pretty sure that I do not tell or get all the names and all the people, but I at least try to figure out some of the names and some of the most important ones, and you will see what I mean. So one of them, first to mention, is Ricardo Melamed. He was a general coordinator at MCTI, and he was in 2010 already on the first workshop, which were celebrated at the UEA on the invitation of the rector there. And Ricardo came just from China in the early morning. He was so tired, but he was there, and he just accompanied Antonio Manzi and me during the next years with all things we had to do at the MCTI. So it was a really great support. And I want to mention Marilene Correa da Silva de Freitas. She's a full professor still at the UFAM, but at that time she was the Secretary of the State of Science and Technology in the state of Amazonas, and then rector of the State University. And you see, these were the connections also to the local or regional politicians already, and the scientific side from the State University. And later on, Marcilio de Freitas, also Secretary of State there, and he's now retired professor at UFAM. And what I would really like to mention is the support, the general support and constant support of Maite, Maria Teresa Fernandez Piedade at INPA. She was a titular researcher at INPA and was a coordinator of this Brazilian-German cooperation over years. And in 2007, the Max Planck Institute for Chemistry stepped into this connection, and with a lot of contacts and a lot of work together. And within these studies all, there were Jochen Schöngard and Florian Wittmann. senior scientists at that time. Now they are full professors at INPA, the other one in Karlsruhe in Germany. But they were my supporters at the institute all the time, and I have to thank all of them very heavily here at this time. So, to get a feeling for all the people who were involved and helped us, just remember, we just identified a site for the ATTO Tower somewhere in the middle of nowhere. We just had a look on some of the maps and said, well, that would be nice. So, but then this question was, how do we get there? What do we find there? How can we transport all the issues, all the things there? And so you see here the map, and it started in 2009 from Bella Vista to do a first identification of the tower site. So you see here the people from Germany who are trying to secure their way back in the jungle. It's not so easy to do that, we learned. And we were supported by people from INPA like Julio Souza, Julio Tota, and then from Conquest Sonda Ltd, and some others from the SDS. And they all were with us in the field, and they did a great job to guide us and to help us to find our way. So easily to be designed, we wanted to go not from Bella Vista. Later on, we looked for another way, and this was again an expedition into nowhere to look how to do this. And this was done by 4 people with Florian Schöngart, and then Hermes Braga Xavier. He was one of the heads in all this organization. And a big thank you to him. All the time you will see some other work and some other support later on. And then Julio de Souza again, Gomez, and some other from UATAMA, Sustainable Development Reserve. And they all were helping us to identify an access to the site where we wanted to see the tower be built. And you see this way already started a little bit away from Bella Vista, and this is where we today have Porto Atto. And so this was the so-called Ramal Paurosa redetected, and we could use part of this old way into the forest, access way to the forest, which was used by perfume samplers several years ago. And so a little bit could be used from this access road already, and but this was of course not the road which could be used for all the equipment. So you see the first transport chains, something like a nutshell for some of the cars and quadros to get there into the forest and to get access and to get forward there. And later on we had bigger cars just getting access within this Paurosa, and I call it Chief Officer Hermes, thanks to him, because he was organizing everything around this access to this unknown area and through unknown roads. And it was really a big work from all these people, and we wanted to see not only the access road but also some towers, and we had some really high classified work to be done within this forest, somewhere in the middle, and we called it Operation Wind Tower in May 2010. This was the building up of a first tower to get measurements there about meteorology and some trace gases. And you see all this work in the middle somewhere, and those people who are working today at the ATTO Tower can imagine, if they go a little bit into the forest, what it meant to do this work there. It was not so easy. And finally, the tower was growing, and the first meteorological tower, which is standing still today, was there. So we got measurements above the forest at 80 meters, and this was already quite high if you compare to the other towers around. Just from the RBR sites, there is no 80-meter tower to my knowledge, so this was really already a tall tower. So 3 operations in June 2010 were ready. This was the access road, was cleared from the shore, so we could use it with smaller cars. We had some soil soundings, and we had an 80 or 81 meter mast for monitoring meteorological measurements already. And we could do a lot of preliminary measurements at that site already to check whether we had made a good choice. And then there were more and more expeditions to check exactly what was going on, and this was the next one in June 2010. You see Antonio Manzi and again James, who was organizing and guiding us. And I'm showing this picture because it was so interesting, because here you see the administration head of the Max Planck Institute in chemistry. He wanted to see where the money is going. He wanted to see what's going on there. And then a lot of people who were supporting this expedition, and the names are here, but I do not want to read all of them. Just see some of them. Rainer Lietz, for example, was of course already there, and Jochen and Florian and Julie Tota. So there were a lot of people supporting all the time. The next story, in the same year, July, August, there were Andy and me within this experimental access again. I'm showing this one because there's one special person, Jeff Tollison, one of the editors of Nature. I'll come back to that story later. He was interested to see what we are doing there, and he made a quite famous sentence, a quite famous publication about that. See you later. Well, end of 2010, there were two towers, because we had already this, as I saw, the wind tower, and Hermes had a lot of work to organize with these people there to build up the today so-called instant tower, again 80 meters, which was also erected in 2010. And now we had two tower systems there, which were really well suited to do a lot of measurements. So we had, at the end of 2010, pilot measurements, which made possible, and we could clarify meteorological conditions and trace gases, and got the first impressions from the site, and we could plan for the next measurements and the next work. And the next work was, of course, the very tall tower. Started already with all this work on the access road, and now we had this February 2008 with Operation ATTO Tower. You see Hermes and Amaury, two key persons during all this time, trying to find their way to the ATTO site, and what not so easy. And you see all this work there. We had the first idea to get a camp, or the first try to get something like a camp to stay overnight. And we had a lot to do with generators. We had a tractor there. We had to build everything. And of course, everything had to be repaired there. And you see all these people there. And it's really remarkable what has been done in the forest with a minimum of technical equipment, and everything was running perfectly, and thanks to INPA and all these people and people from around in the forest who supported this work. It was incredible. Then August 2012, the real professionals from outside came. This were to build up this bigger road to get a good access, and this was the state of the Amazon who organized or paid for this. And this was called the opening of the Ramal ATTO. And today, when you get to the ATTO, you can have, if you compare it with this, you can have the feeling that you're running on a highway. So it was easy to show. 2013, we had a campsite with a kitchen, with a dormitory, with a watchdog, of course, and I was very happy. And so it went on. In August 2014, we had already the laying of the foundation stones. You see, at that time, the INPA director, me, was trying to play a little bit around like kids with this machine, while Najibi was already caring for everything to get food into the camp. And August 2014, the official laying of the foundation stone, a lot of hats were coming there from Germany, from the MPIC, then from the UEA, INPA director, and from the Max Planck Society, Andreas Trepte, and from Sun, the company, Sergio, who was the chief, the chef of the company to build up the tower, and Hermes involved all the time, and with all his knowledge about our wishes and the possibilities of the technical background there in the things. And there are a lot of other people. Arji, who was cooking so well for all of us. And these pictures showing Ari at his work in the kitchen and with the big fish there. But what I would like to stress is Arji or other people, they were not only doing one job, they were always doing several jobs. So they were really getting out, helping to build up had to build up something which was needed and to care for everything. And this is really remarkable for all the people who were supporting us there. Also for Amaury, was there for electricians, for all the electrical parts, but he was going directly to the generators and repairing generators and everything else would have his hands on. Andre in the kitchen and on the engine of the truck. So all the people doing several jobs, and Antonio, Madruga, they're always caring for everything. And this was really something which was running ATTO, because everybody was involved in everything and everybody helped or worked whatever was needed. And this was well thanked by all the people, all the scientists like here, because this was always a welcome and everybody cared that the ATTO project was running forward. This was next step. Rainer discussing with Hermes about the current situation and the planning for the next steps in ATTO, and you see they were behaving already quite similar, Hermes and Rainer, and so these were really a cooperation team and caring for everything, caring for repair, like here, then trying to fix all the notes and all the remarks, just to tell and to discuss with everybody and discuss with Najibi about all the plans and checking with RG and helpers in the kitchen to chair, to see what was doing there, what has to be ordered and what has to be repaired. And in the evening, joining all the discussions with scientists at one table and to see what has been done over the day and what has to be planned for the next day and days. So there was a lot of information spread by this guy, and he was organizing everything. And he was also organizing one of the famous visits of the German ambassador, Georg Witschel, visited ATTO in March 2017. He wanted to see ATTO and to see he was coming by helicopter because this was more comfortable for him. But by the way back, he took the choice. He chose the way by taking our boat because this was of higher interest for him than going back with a helicopter. So he was thanking Hermes for his work there, organizing the trip and all the things around ATTO. And then Hermes brought him back to the river, and they took their way back to Manaus. And then, of course, they had to relax. And it was wonderful to see how everything worked. And it was a towering experiment. And here I come back to this Jeff Tollison, the editor of Nature, because he was writing a paper when he was joining our expedition there. And he said, well, it's an ambitious project. And it will provide crucial data, will be wonderful. And then he said, but only if scientists can get it built. So I translated here by Google into Portuguese for those people who speak better Portuguese. So he had some doubts, but there were no doubts because already the Sun people said, yes, we can. And you know, the tower is ready today, it's working, it's operational. And this picture of all these small snapshots may give you an impression. This is only a small number of people who were doing a lot of work around the ATTO tower to build up this ATTO tower and bring it in these special, give us the situation to do these measurements there. And thanks to all the people and all the unknown people, all those people where I have no pictures from, but it was a lot. So we got it built, but not only the scientists. There was a lot of other support, people from the surrounding areas, people from INPA, people from, well, all over Brazil. Some people came and helped. So let's celebrate. Some of the people now getting into pension, some retire as me, and so they have to think about what they do now. And I think we took some pictures, or one picture from Hermes at the Lake of Malbina. And I would say to Hermes and to all the other people, enjoy your time and thanks to everybody who helped us all the time to do this wonderful job, to build up this tower here, to build up the campus, to give us the possibility to do science and to get these wonderful site reports by Rainer. And so, obrigado, I must say, muitas outras. It really was a wonderful support all the time, and thanks to all to give us the chance to work here in the forest. Thanks.
Thank you, Jürgen, for this nice overview into the history of ATTO and all the people that was involved in the construction. And now we welcome also from the INPA, a doutora Ilândia Brandão de Cunha. Ela também vai a falar algumas palavras. Ela é a doutora de ciência do Centro de Energia Nuclear, a atual diretora substituta do INPA. E atua na área de ecologia. Também é especialista em balanço de massas e recursos hídricos. Muito obrigada.
Primeiramente, gostaria aqui de agradecer a coordenação da doutora Susan e do doutor Cada, ao nosso colega Hermes Braga Xavier. Dizer que sinto muito orgulho de com todo. Ok. Nos conhecemos de longa data, desde a antiga coordenação Ciências do Ambiente, lá em 1987. Depois, o Hermes recebeu o convite do Dr. Antônio Nobre para integrar a equipe que implementaria o programa LBA. E desde lá, o Hermes vem desempenhando seu trabalho com muita dedicação, empenho e profissionalismo. Quando os idealizadores do projeto ATTO pensaram em instalar uma torre em uma região praticamente remota, estava lá você disposto a integrar a equipe que tornaria esse projeto uma realidade. Se hoje temos a nossa torre instalada e instrumentada para que todos nós possamos desenvolver nossos projetos na busca de novos conhecimentos sobre os impactos das mudanças climáticas na região e a influência da floresta no clima global, é graças ao trabalho de pessoas como você, que ajudou a concretizar este lindo sonho. Parabéns por ser este profissional exemplar. Gostaria aqui de registrar nosso reconhecimento da importância do seu trabalho no desenvolvimento ao longo dos anos na Torre ATTO. Hermes, receba esta homenagem como símbolo do que você representou e representa para o desenvolvimento desse projeto. Que merecida. O INPA agradece por ter servidores como você comprometido com a instituição. O nosso muito obrigada a você por fazer parte dessa história chamada Torre ATTO. Mas, por favor, receba aqui. Eu fico até emocionada. Mas eu gostaria aqui de passar em suas mãos o certificado de apreciação orgulhosamente entregue a você, em homenagem a todos os seus anos de dedicação ao trabalho inestimável ao projeto ATTO. Receba aqui, em nome da coordenação de todos os que fazem essa família que se chama Torre ATTO, um agradecimento para o seu esforço ao longo desses todos esses anos.
Muito obrigado. Oi. Muito obrigado. Agradeço a confiança que me foi depositada na época. E só tenho que agradecer. Foi um desafio, todos sabem, foi grande o desafio. É uma região inexplorada, chegamos lá do zero, no meio da floresta fechada, e demos início a esse grande projeto. Para mim foi um grande desafio, o apoio da minha família foi fundamental, dos amigos e do grupo todo que estava com a gente. Foi uma determinação para concluir esse grande projeto. Muito obrigado.
Okay. So thank you to Hermes and to all the people that make ATTO possible. No, not only scientists, as we heard from Jürgen, it was many people involved.
I guess, it's quite emotional. But if you allow me, I will speak in Portuguese. É muito emocional, né? Acho que todo mundo ficou emocionado. Eu fiquei. Eu e a Su pegamos o trabalho fácil, né? De pegar a coisa toda construída. A gente só chegou para cuidar, né, Susan? E o Hermes, outras pessoas também, como o Jürgen, mostrou muito bem, mas o Hermes é fundamental. Está indo agora buscar outros desafios, um lugar que ele, né? E mais essencial, sabe? É muito fácil a gente pensar em chegar hoje no ato, encontrar toda aquela infraestrutura, mas eu já ouvi histórias do Hermes contando no campo que foram fantásticas, né? Deles carregando os pedaços dessas torres nas costas, no meio da floresta, sabe? Então, pensar onde nós estamos hoje, graças a pessoas como Hermes. Então, muito obrigado, é pouco. Obrigado, Hermes.
So just to translate a little bit, Beto and I got the easy job. We came on so I think the hard job was, the dream, building the tower, and this was, this was Manzi, this was Andi Andrea, this was Jürgen Kesselmeyer, and all the team people like Hermes who really had to build zero this amazing infrastructure that we have now. And we came at the end because it took so long to build that it was so hard to do, and really it's an amazing accomplishment to have such an infrastructure in the middle of the forest. And I remember when the Minister of Science and Technology visited earlier this year the site, and nobody could believe how amazing the facility is, all these things. It's just an amazing achievement. So we realized that not only Hermes is leaving, but also soon one of our other people will leave the project and retire. So we wanted also, not to steal anything, but it's also the people who work on these tall towers. They're not, that's also to put the instrumentation, to design, how the instruments will fit on them, how to move the air from 300 meters to the bottom, and without having condensation and things happen on the way. So we also wanted to just take a minute to recognize Olaf Kohle, who's the head of, from the German side, the tower team. And so Olaf will be retiring later this year.
Thank you. I didn't expect it.
So he was part of, yeah. Okay.
Thank you. Only to be here and to work on this big project and to contribute so that everything will come to a good end. Thank you very much. And yeah, and now, of course, I will retire next year, and so will not have the chance to come back anymore, maybe. And Tarek will be my successor, and I think he will do also a very good job here.
Take a picture. Yeah, so maybe two photos. So I think it shows that we need everybody for this project to work. So, and we're thankful for everybody, and we'll all move on someday.
Okay. It's time for lunch now, and so we have to thank everybody that online. Thank you, Jürgen, for your presentation. Thank you, all the people that were present online. Now we make a break for lunch to get strong for the afternoon.
Okay, and also the posters that you saw this morning, and all the posters are in this Gather Town environment. So if you don't remember how to use it, maybe we can give a tutorial a little bit later. The posters are there, so go take a look.
They're coming. Oh, look at that. Okay. Okay, so I think we should start to keep on schedule. So, hello everyone, good afternoon. I hope you have had a good lunch. And I'm also happy to be here to discuss a little bit more about science at ATTO. So I'm happy to start the first, let's say, in-person session, poster session. So I think the idea is to discuss in a more informal way what we have been doing at ATTO. As far as I remember, this is the first of 3 or 4 sessions that we will have over the week. And its name is Ecosystem Process. So you can imagine from the title how broad this thing is. But I think it's very nice because it's super representative of what is going on in ATTO. I think we have everything from molecular level until the landscape. And I think that's exactly what we should be doing, or at least one of the things we should be doing. So nice to see that we have this up-down scaling. And I think many of us here is, at least in this session, is trying to help a little bit with this challenge of combining tower satellite data with ground data. So that's at least one of the most important tasks as we see in our group, as you will see in the posters we have in the session. So yeah, I'll be happy to discuss with you about these topics. So I don't know,
yes.
Flavia, if you want to say something.
For coming here to our section. And now we are beginning the PICO presentation. 3 minutes to each poster that you can explain to you what the poster is talking about. And after that, if you are interested to the poster, you can go to talk with the people in the section.
Okay, so we are 15 people, so quite a lot. So I would like to ask those presenting here to get really, really to pay attention to the time. So to be quick, I think we will have enough time, and it's better if we have time to discuss this more informal. So I am the first of the list, and I don't have slides, but I can tell you what my poster is about. It's basically going in this direction of merging satellite with field data. So we were basically working with different scales of satellite or remote sensing measurements, using both drone acquired images, but also satellite, especially Landsat and a little bit of Sentinel. So merging this with ground data to try to understand a little bit how does water seasonality and also soil attributes change functions of forests across this nice topographic gradient that we have at ATTO. So to make this, we basically installed several plots over different drainage lines that we have around the camping, where we started to monitor the trees. We're measuring trees greater than 20 centimeters dBH, because those are the trees we can see from above with the images. And we go measuring a bunch of variables of these trees. So since diameter growth, soil moisture, also apart from measuring growth with tapes, we also go installing automatic dendrometers. And taking or monitoring the plots with a multispectral sensor to acquire, apart from automosaic data that allow us to produce digital surface models, but also to assess, for example, canopy temperature, water content, and so on. Yeah, so please come to see the post. Next.
Next is Ianara Ferreira.
Ianara.
Slides número 2.
Boa tarde, meu nome é Yanara, eu sou estudante de doutorado do Instituto Nacional de Pesquisas da Amazônia e o tema do meu pôster aqui no workshop é investigando o comportamento mecânico de árvores amazônicas. Estou mais ou menos nessa sessão 1.2 e quem quiser ir lá para a gente trocar uma ideia, conversar, será bem-vindo. Bom, vou dar uma contextualizada do que eu estou trabalhando no momento. A vulnerabilidade de árvores e chuvas e ventos extremos, elas podem ser influenciadas por variáveis ambientais, traços funcionais das espécies e biometria dos indivíduos. Sendo assim, na floresta amazônica, em florestas tropicais, os danos e mortalidade de árvores por chuvas e ventos extremos, eles geram emissões de carbono expressivas. A ação desses ventos nessas regiões, elas podem causar a desfolha até a quebra de troncos e destruição do dossel. Assim, o meu objetivo nesse trabalho é analisar o comportamento individual e de grupos mistos na interação com ventos e chuvas extremas. Aqui é mais ou menos para contextualizar onde é que eu estou trabalhando. Estou trabalhando O meu projeto, ele é um estudo do projeto Inventa ATTO. A minha área de estudo está localizada na Estação Experimental de Silvicultura Tropical do Instituto Nacional de Pesquisas da Amazônia. Ela é mais conhecida como ZF2 e é uma floresta de terra firme. Essa área foram instaladas 3 torres em diferentes perfis topográficos, na encosta, no platô e no baixio. Essa minha coleta de dados é com base nessas torres instaladas e também em sensores que vão ser instalados em árvores. Aqui a gente pode observar mais ou menos como é que são a distribuição das torres. Elas têm aproximadamente entre 33 e 36 metros. E nessas torres foram instalados instrumentos como o anemômetro e o pluviômetro para analisar variáveis de vento. Elas têm como, para coletar os dados, são utilizados baterias e painel solar. E é isso.
Luciano?
Boa tarde, todo mundo. Meu nome é Luciano, sou estudante de doutorado do INPA, sob orientação do Daniel Mar. Vou apresentar rapidamente os resultados gerais da nossa pesquisa, intitulada Sensibilidade de satélites ópticos na detecção da mortalidade de árvores causada por windfalls, aqui na Amazônia Central. Nosso projeto ele é focado também no upscale, ou também na detecção, na integração de dados de campo com dados de sensores remotos, né? No caso, nossa pesquisa é específica e voltada para impactos causados por windfalls. Então, e de muita importância, porque windfalls... Onde é que está o laser? Aqui. Windfalls são uma das principais causas, né, de mortalidade de árvores. E as ferramentas de sensoriamento remoto têm nos permitido há muito tempo monitorar esses eventos e extrair métricas de distúrbio. E nós temos métricas que são muito sensíveis e possíveis de serem detectadas por sensores remotos, como, por exemplo, o percentual de árvores mortas. Então, nossa pesquisa está focada em responder 2 questões. A primeira é como a resolução espacial de diferentes satélites afeta a detecção desses eventos e a extração dessas métricas de mortalidade de árvore. e qual sensor produz estimativas mais confiáveis, para a gente poder trabalhar, por exemplo, em extrapolações em nível de paisagem desses eventos. Então, aqui é uma imagem de demonstração das diferenças de pixels entre os satélites utilizados, né? Então, para dar uma ideia da magnitude das estimativas que nós estamos apurando. A nossa área de pesquisa ela é também na Amazônia Central, próxima à cidade de Manaus, né? Teve um evento de mortalidade, né? Um blowdown, a cerca de 40 quilômetros aqui de Manaus. Então, nossa pesquisa ela combinou, primeiro, instalação de parcelas, levantamento de árvores vivas acima de 10 centímetros de DAP, né? Então, combinando abordagens de inventário florestal, né? Combinando a abordagem de sensoriamento remoto. Então, nós utilizamos 3 imagens de diferentes satélites, de diferentes resoluções espaciais, para imagiar esse distúrbio. Utilizamos análise de mistura espectral para extrair as frações NPV, né, de vegetação morta, e combinamos a modelagem de campo e integração com dados remotos para poder criar essas métricas e avaliar como esses diferentes satélites estimam a mortalidade de árvore. Então, é isso. Nós temos diversas métricas, nós temos resultados diferentes para cada satélite, e eu espero poder conversar com vocês mais. Nosso pôster está ali para frente. É isso. Obrigado.
Adriana Peixoto.
Então, a Adriana, ela não pôde estar conosco, mas nós temos o pôster dela, né? Bruce, não sei se você quer falar alguma coisa. A gente pode conversar sobre o trabalho da Adriana. Na verdade, é um dos... Sorry, I was speaking Portuguese. I just switched because of the presentations. So Adriana could not be here with us, but she sent her poster, which basically belongs to her PhD thesis. She's been working with us now since maybe, I think, 3 or 4 years already. And for her PhD, she's working with me and with Bruce. And one of the things she's trying to do is basically to combine all the expertise, all the experience that Bruce acquired with different colleagues by studying this phenology of the trees using his mounted camera that is already in ATTO since 2015, right? Right, Bruce? Yeah, so in Adriana, she's basically learning with Bruce, how can we use this knowledge, how can we apply this knowledge basically when we have, for example, drone imagery cover much larger areas. So can we also get some numbers on the phenology stages of the trees, for example, around ATTO, going beyond the plot that Bruce has been monitoring since a while. So the project is basically about how can we set the color of the images that we acquire on the drone, that they can technically represent what we think or what we want they are representing. So Bruce will explain a little bit better than me, of course, during the poster session, so please come to see. I think it's very interesting. It's the physics of the pictures. Yeah, next.
Caroline Malman.
Bom, boa tarde a todos. Meu nome é Caroline Malman, sou aluna de doutorado da Universidade Federal de Santa Maria. Em parceria, em colaboração com o ATTO, estou desenvolvendo então um projeto que são aplicações de sensoriamento remoto ao longo de um gradiente hidroclimático em ecossistemas amazônicos. Bom, em tempos, então, né, de grandes desafios para a conservação de ecossistemas florestais, a gente tem que utilizar abordagens que nos permitam analisar em múltiplas escalas e também em fontes de informação contínuas, né, para fins que a gente consiga atingir objetivos de conservação. Nesse sentido, o sensoriamento remoto, ele permite que a gente possa extrair informações biofísicas e bioquímicas da vegetação para fins de monitoramento, através de técnicas combinadas como de espectroscopia de campo e de imagem. Então, buscar esses novos conceitos e novos desafios para que a gente consiga aplicar e integrar esses dados in situ a dados de sensores orbital é uma busca dentro do campo do sensoriamento remoto. Nesse sentido, a Leaf Mass Area, que é a área foliar massa de área foliar, né, LMA, ela é um importante variável e um importante bioindicador nessa variação desses processos ambientais em questões de espaço e tempo. Também é um potencial bioindicador, né, das questões de estresse hidrológico e ainda corresponde a um parâmetro-chave de entrada para modelos globais de vegetação, de dinâmica da vegetação e balanço de nutrientes. Portanto, o principal objetivo do projeto, junto ao ATTO, é analisar a variação espaço-temporal da LMA em um gradiente hidroclimático, utilizando técnicas de sensoriamento remoto em múltiplas escalas. Pode passar. Então, por meio de métodos então combinados de espectroscopia de campo, espectroscopia de imagem, associados a traits foliares levantados em campo e a variáveis climáticas, a gente vai tentar responder essa pergunta. E entre os resultados esperados é desenvolver um modelo empírico e um modelo híbrido, né, para estimar e recuperar a LMA em escala regional, em nível de ecossistema, principalmente nos principais ambientes de terra firme e floresta alagada, e também comparar a robustez e a capacidade de generalização dos modelos em diferentes escalas espaço-temporal ao longo desse gradiente hidroclimático. Obrigada, fico à disposição no banner para mais informações.
Gerd, you are next, please.
So hello everybody, thanks for still being here. We are working on a project for a longer time, which we started several years ago with several people from IMPA, mainly Beto and Savio, and with some new people from Piracicaba, looking a little bit for the microbial community. And Carsten, who started with us, is now in Leipzig, and he still continues to work. And what we plan to do here is to develop a new method how we can follow processes which are happening in the soil over time and to understand what the system is doing. And in order to do this, we have three main players. We have plants in the system. You know, they make leaves which are falling down on the ground, but there's also exudation, and there are microorganisms which decompose this material, and there is soil where we have a lot of sorption, desorption going on, and different spatial characteristics. And in order to look at this, we focus on what is dissolved in the soil. We have this dissolved organic matter. We can find something which comes from plants, something which comes from microorganisms, something comes from the soil. And if we analyze this material, we can say what is going on. So is litter decomposition running, or is it more exudation, what we see, for example? But in order to do this, first, if you don't know what dissolved organic matter is, here are some pictures not from the Amazon. As you see from this picture, it's winter in Germany, so a lot of snow. And you can see this brown stuff which you see here, and it's decreasing with depth. This is the dissolved organic matter which we have. In summer, it's still brown, but you see when the temperature goes up, there is much more decomposition in the system. But now we need also something to look at this material, and what we use for this is a very high resolution mass spectrometer, and you see here the ion source, so there's a spray of vapor, and you charge the molecules, and then you can weigh them in the mass spectrometer. And the machine which we have has a very, very high resolution. So what you see is here's a mass range of up to 1,000 Dalton. So a molecule weighs typically around 100 to 300 Dalton. Yeah, you can see from the size distribution how many molecules we have. These are thousands of molecules. And if we zoom in a little bit in this picture, yeah, you see here, these are now 110 Dalton ranges. This is a single Dalton. Yeah, and this is a zoom in a single Dalton. There are many, many peaks under a single Dalton, and as our resolution is extremely high, yeah, you can see here the digits behind the comma, and all atoms have a strange number, I would say. So it's not, the mass only for carbon is 12.00. There are always difference in the digits. We can calculate what is the structural formula, the elemental formula. This peak, for example, with this mass has 15 carbons, 15 hydrogens, and 8 oxygens. And this helps us to say which kind of molecule is this what we are looking for. And if we apply this, for example, to soil and look how much of the variability in our samples is explained by which factors, we see that around 25% of the variability comes from the microbial community on what they do, 20% comes from the soil, and 10% comes from root. And the interaction between microorganisms and root is very big because we have a strong overlap of about 13%, which indicates that molecules which are decomposing from litter or which are exudated are in close contact to the microorganisms. They exchange a lot. Yeah, and in total, these are more bacteria which are doing this. And we have 2 PhDs now running, one more on the ecosystem scale and one more on the microbial scale, and you will see their posters now. Thanks a lot.
Frederick.
So this is Frederick.
Okay, hi everyone. My name is Frederick. I'm a PhD student at the Friedrich Schiller University and the MPI for biogeochemistry in Jena. And so, as Gerd already introduced, my side of the project is about dissolved organic matter cycling in soils of different Amazon rainforest ecosystems. And so here at ATTO, we are using 4 main sites for sampling, which belong to 2 major types of forests around ATTO here. So these are 2 sites with terra firma type forest growing on clay soils, and we have one site on the plateau and one site on the river terrace. Additionally, we have 2 sites which belong to the type of white sand forest around here, with one Campina and one more Campina rana type forest. And at each of these sites, we are sampling soil pore water on soil profiles from 0 to 30 centimeter depth. And we are using this to investigate and measure the water chemistry and the molecular composition of dissolved organic matter, DOM. And we have been doing this since 2018 with roughly biweekly sampling, which was always done by colleagues from IMPA here. And so by now we are in the process of creating relatively long time series. And so we do this because we want to learn about the cycling of DOM and about understand processes of organic matter input, decomposition, and transformation. And specifically, we want to know what the differences are in functioning between these different forest types. And I want to give you just a very general idea of what kind of results we get from our analyses. So this plot that you see, it summarizes major variants in the molecular DOM composition data. And the only thing that I want to point here is that you see 2 distinct clusters, samples belonging to the white sand forest on the left side and samples belonging to the terra firma forest on the right side. And what this shows us generally is that the DOM composition differs very strongly for these 2 major types of forests around ATTO. And these differences are indicative of a highly different extent of the cycling and processing and decomposition of organic matter in these soils. And so if you want to learn a bit more about how these systems are functioning in terms of organic matter cycling, please meet me at my poster. And lastly, I just want to say thanks to all of the co-authors and all the people who over the years contributed to making this work possible. So thanks a lot, and thanks for listening.
Thanks. Next is Jessica.
Hello. So, waiting for the slides. Oh, perfect. So, hello everybody, and welcome to my short Pico talk about the enigma of microbial community composition and functioning in different Amazon rainforest ecosystems. But why should we even care about microbial communities, how they look, and what they do? So, as most of you hopefully know, within the Earth's surface, the critical zone is very important. It is a major interface of biogeochemical processes. For example, the carbon cycle, but also the water cycle. And within this critical zone, we have dissolved organic matter, DOM in white, and we have also volatile organic compounds, so VOCs. And these are important because for dissolved organic matter, they mediate and affect biogeochemical processes, while the VOCs affect atmospheric processes. And especially in the Amazon and other rainforests, this is important for the Earth's climate system. And what can we learn from them? So the DOM and VOC, as I said, are important signal molecules, as Gerd already said, that mirror and reflect biogeochemical processes. And these molecules are highly intertwined with the microbial communities, so bacteria, archaea, and fungi. So why are they? Because they can consume, produce, but also transform these molecules. However, we don't really know much about these interactions the microorganisms have with DOM and VOC. And if you want to know more about what we already know about these interactions, or what I plan to do within ATTO to tackle the question of these interactions, please come see me at my poster.
Ilana Hadlich.
Só passar aqui, né? Boa tarde a todos. Meu nome é Ilana, sou aluna de doutorado da Botânica, aqui no IMPA. E vou apresentar para vocês o trabalho então, que a gente está fazendo no sítio do ATTO, sobre variação de caracteres funcionais e o reconhecimento de espécies arbóreas em diferentes hábitats da Amazônia Central. Bom, as mudanças climáticas e a rápida perda de habitats estão causando a extinção de espécies e mudanças na composição e estrutura da vegetação, o que representa grandes ameaças à biodiversidade, no qual ainda não chegamos nem a metade das espécies a gente conhece aqui na Amazônia. São registradas atualmente 6.000 espécies, mas com uma estimativa de 16.000. Então, nesse sentido, entender a vulnerabilidade dos diferentes ecossistemas e das espécies se torna de grande urgência. Nesse sentido, nós conseguimos avaliar a vulnerabilidade das espécies tendo acesso às características funcionais. Assim, a gente consegue avaliar estratégias ecológicas das espécies e vulnerabilidade à seca. E, além disso, o desenvolvimento de ferramentas que nos auxiliem a agilidade nos estudos ecológicos é um grande avanço para a conservação. Então, nesse sentido, a gente tem utilizado a espectroscopia no infravermelho próximo, que nos auxilia tanto na identificação de espécies, quanto na determinação de características funcionais. E, então, com isso, o objetivo do trabalho é avaliar as diferentes estratégias das espécies arbóreas mais abundantes entre terra firme, campinarana e igapó, lá no sítio do ATTO, e avaliar o potencial de espectros NIR de tronco e folhas em campo para agilizar os estudos ecológicos aqui na Amazônia. Qualquer coisa, estou à disposição. Obrigada.
Now is my poster. Okay, my name is Flávia Durgante, and I'm a postdoc researcher in the Karlsruhe Institute of Technology with Florian Wittmann and Maria Teresa Piedade and Jochen Schonger. Our group work here in IMPA with Mauá group. And now I will show you some results that we're monitoring the water source and the xylem water from 1 year. And our poster today is talking about how we are trying to understand the vulnerability of different ecosystems, analyzing the source of water. Yes, we monitoring the rain water and soil water, rivers and lakes, and analyzing the oxygen and hydrogen isotopic signal. We built the local meteoric water line to ATTO site using 3 years, and samples are collected weekly. And now we have our line that we can compare with the water from the xylem to see if we can recognize the source or what's happening. Yes, and we used 5 sites, 3 flooded forests, and 1 plateau area, and 1 Campina Arana that has sand soil. And we can see that the xylem water, we analyzed the xylem water from monthly, from 1 year. And we can see the xylem water are evaporated, because in black is our local meteoric water line. And this point, it's the xylem signal, and the red points, it's the dry season. And we can see the flooded forests are more evaporated than terra firme, and maybe is more vulnerable to extreme drought years. And I have more details in my poster if you want to see there. You are welcome. Thank you.
Gisele.
Hi, good afternoon. I'm Gisele. I'm a postdoc researcher at the Karlsruhe Institute of Technology and also Mauá Group. And I want to invite you to visit my poster today that is about the project I'm working on that is looking for different functional traits of tree species at the whole plant level in different forests in the Amazon along a climatic gradient. And, well, because climate change is going to affect the tree growth and survival, especially through change in water availability, we want to investigate how tree traits and strategies change in different hydrological regimes and across different forest habitats. To do that, we're going to use a very interesting data set with 4 research sites. One includes the ATTO site, and the other 3 ones we are going to establish along a climatic gradient that is a gradient of precipitation that has an increase in the dry season length. And we are looking for 3 forest habitats, the terra firme forest, the seasonal flooded forest, and the white sand forest. This is quite interesting because most of studies don't include the diversity of the Amazon habitats. And I'm going to look specifically for some leaf traits that are related to the fast slow strategy and plant hydraulics of the most dominant tree species in these habitats. So visit me in the poster, and then we can discuss more about our main questions and main hypotheses. Thank you.
Priscila.
Good afternoon, everyone. I am Priscila, and I will be presenting the research work entitled Monitoring the Tree Radial Increments in Different Phenological Groups at the Terra Firme Forest in the ATTO site. So, for this research, we choose 122 trees from the Terra Firme at ATTO site for install the dendrometer bands, and we go there monthly to measure their growth. And in addition to this, these trees are daily monitored by a phenocam. So I hope to see you at my poster. Thank you.
Ingrid.
Hi everyone, I'm Ingrid Chanka. I'm a postdoc at the Max Planck Institute for Biogeochemistry in Jena. First of all, I'm sorry I don't have slides. I got a problem with my computer and I was fighting, but there was no way. So yeah, just a quickly summary of my poster. As we saw in this morning, the presentations, there's a lot of discussion on the role of ecosystems as a net or sink of CO2. And related to this question, we also have the question of how long carbon stays in the ecosystems, right? And to tackle this question, I'm going to present my poster, an approach to estimate the mean transit time of carbon in the Central Amazon based on measurements using the Stand Tower in ATTO. So the transit time is basically the time it takes carbon to be, after it's fixed through photosynthesis, to be released through respiration. And by measuring CO2, radiocarbon in CO2 and CO2 concentrations in different heights below the canopy level, we use on a mixing model where we estimate the radiocarbon signal on the ecosystem respiration, and with this value we obtain an estimation of the mean transit time of carbon. And if you want to know more about it, how we do that, which kind of measurements we got and everything, please come to my poster.
Okay, so seems like we are done. I think it was quite good in time. That means we have enough time for the posters, right? So I think the idea now is that we bring some stands where we will hang the posters here in the auditorium. So please correct me if I'm wrong, but I think that's the idea. Yeah, so, and then it's, again, like a real conference where you go around, you talk to people. Question? Someone has a question, something to add?
Yes, I want to say thank you for everyone to coming to our section and I hope to see you in our poster.
Cool.
química de compostos orgânicos voláteis biogênicos pode ser emitido pela serrapilheira
can handle the mic. And the mic, yeah. This thing at least. Okay, welcome everyone back to the last session of today. Thank you all for holding off on the beer a little longer, and then we can go start with the beer. Today, the last session is about greenhouse gases and VOCs. And as you can see, we have quite some talks today. A few people couldn't make it. Sylvia's not here, but her poster you can find online in the Gather Town. Murielli is also not here, but she will present the video. So the last thing is a video, and if there are questions, you can also talk to Eliane. And Itiara, I'm actually not sure. Is she here? No? Okay, then we have only a few talks today, and we'll try to keep it short because, of course, beer is waiting. So we start our session with Oscar. The next slide. Yeah, you can already try that.
Balloon. Oh, very welcome. Hello, my name is Oscar. I'm from IPEM, São Paulo, and I will talk about the biogenic volatile organic compounds fluxes in ATTO site. Well, first we have to learn about the VOCs, what it's for. Those VOCs are gases that liberate from the leaves of the trees. And they use the trees to communicate, each tree, talking trees they call. And also they communicate with bugs and communicate with us. But we don't understand yet what they are doing. That's why we are studying hard. I will give an example of that. Like VOCs of terpenes, monoterpenes. One calls the limonate. Limonate are molecules that chirals are like two hands. The right hands and left hands looks like same, but are similar, not the same. And these hands, like limonade on the right tastes like lemon, but the left tastes like orange. So you see, are different. Eh? So this is not our worry about that, but we are working with the VOCs and fluxes. Why fluxes? Fluxes because these VOCs are liberated from the leaves, as I say, chemical atmosphere that goes to the aerosol, SOA, secondary organic aerosols. These aerosols became clouds, and clouds, it comes rain. That's worry for us, because I'm from Paraguay and South Pablo and all this South America. We receive all the clouds forming here to rain there, or sometimes not. It's dry. This is our worry. But how do they use the VOCs? VOCs are measured with the REA, relaxed eddy accumulation. The REA, the relaxed eddy accumulation, is an instrument that collect the in cartridge, and those cartridge that has chemical absorbent. Then we use in a laboratory. You have to find out what type of compounds, chemical compounds are. So it's a slag time, that too much time. There's an instrument better, PTRMS, but PTRMS is heavy, big one, like Greg says. It's a big, complicated to see mass spectra, you know. This REA is easy, but has problem as well. It's easy because it's portable. You can carry on up to the tower. It's cheap compared with the PTRMS. So Jonathan Williams from Max Planck said Vox are stars, Vox stars. So I said Vox stars is one of my students, Carolina Osterman. She's here. She developed in California the new REA. The new RNA is more portable, are smaller. It's a real time. He can measure it right time, and also can send to the cloud, but not the cloud it rains, the cloud the internet, you know, it goes like that. Anyhow, so we are developing a new REA that Carolina will speak, but there's another problem with the REA. to measure the standard of the fluxes. Like Susan Turnbull said, how to measure the flux from the bottom of the tower to the top of the tower, 325 meters. It's not easy. So we find one is radon. So this is Matthias Sogveld and Santiago, who is here. We were collecting soil in the tower, in the ATTO tower, and we find out uranium and thorium. Thorium releases radioactive compounds is radon. The radon is really nice isotope because radon-220 is like our watch. Every 58 seconds decays in a half-life. So you can measure in the tower how fast it goes, the radon. This is that. Theoretical, but we have to do that. Sometimes this instrument doesn't work, and this is work with another book star, my student Bruno Tapis here, with other Professor Paulo will talk about. Well, in resume, we have to understand the box, to understand what this house that we live together, the earth, we have to take care of that. Thank you.
Okay, thank you very much. And we're now to the next short talk from Carolina.
Só apresentar um da introdução. Obrigada. Oi? No pointer? Acho que não precisa não, Mariana. So, hi everyone. I'm Caroline Osterman, a PhD student of Professor Oscar Vega from IPEM USP. And I just started my PhD program in October last year. So I'm just show you some hypotheses and a motivation. For this PhD, I have a motivation to build a device that I can see PVOC flux in a long-term period and online. I was inspired in the older system, the older REA system, that I worked during my master's degree. The old one, it is this first box in the left, in the right, sorry. And so I developed the new device in the University of California, Irvine, with Professor Alex Gunther lab. This new one, it is a REA system coupled with a GCPIG. I guess I need the pointer. So this is the new REA system, and this is the GCPIG that I can attach in this one. And when I run, it is online, and I have some flux results after 50 minutes by 50 minutes. And you can see all flux results in a remotely way because it's a connect Wi-Fi server. And this, I don't know, the picture is so small, but this is the anemometer that I use. And for this, I have some previous campaign to collect some data about isoprenes, concentration, and I did the night collections with this pump, and during the daily collections in acai plantation, and my intention is just compare what's happened with the acai plantation during the day and during the night with the Amazon forest. This is a pristine, with this new device that I validate with a PCR mass from Professor Alan Gonstein in the University of California in Berkeley. I don't have any results yet, but I will do the first campaign in February next year. That's it, guys.
Bruno? Uhum. Sim, mas só essa, não? Ah, dessa vez. Ah, tá. Aí depois já
Hello everybody, my name is Bruno and I am a student of the University of São Paulo. Oscar is my advisor and the person that is talking after me is my co-advisor. So in our worker, we access the activity concentration of 238 uranium soil samples collected along the ATTO site. The 238 uranium produces in its decay chain the 222 radon, which is a tracer for atmospheric transport studies. 48 samples were analyzed using the instrumental neutron activation analysis technique, and the content of uranium was converted to activity concentration of 238 uranium. Despite the small sample size, the PCA, the principal component analysis, made using novel metals indicates that there is significant difference in the elemental composition of the soil of Plateau, Campina, and Terrace. This is important for my study because it indicates that there is different uranium content along the ATTO soil, how we can see in the box plot. So in the future, 226 radio, 238 uranium will be accessed in more soil samples. Also 222 radon will also be accessed at different heights of the Insta and ATTO towers, thus making possible to understand the behavior of this gas along the ATTO site. Thank you everybody, and feel free to after to make some questions in front of my poster. Thanks.
Thank you, Bruno. Okay, now the next person, Paulo.
Hello everybody, my name is Paulo Cardoso. I'm from IPEM too. IPEM is located in São Paulo, inside the São Paulo University. And I started working with radon measurements last year in November. It was my first time at ATTO. So we still have a short period of campaigns. In fact, for radon measurements, you have just one campaign results here to present. So we could not have any results or any conclusions about this. But the intention is to continue measuring from time to time. And we are measuring radon using an instrument called HAD7, that is an instant measurement. And we also use CR39, that is a detector that integrates the case of radon for a period of about 3 months. But these results we don't have yet. So I'm presenting for you in the poster what we find in this last campaign, the last measurement that we did. We are measuring radon in soil and in air, and it was measured in the 3 different places at ATTO, in Plateau, in Campina, and in the lower part next to the river. And I invite everybody that wants to know any more to see my poster. Thank you.
Okay, thank you, Paulo. The next talk is from Eliane about her poster.
Hello, everyone. I'm Eliane, and today I'm going to present, summarize what our work package for the ATTO Plus. So the title of this work package is Volatile Organic Compounds, VOC Emissions from Amazon Forest, an Integrative Approach from Soil to Atmosphere. So Oscar did already a good job, so I don't really need to go into this, but this is just to illustrate that VOCs are very important for biosphere-atmosphere interactions. They are important mediators, and we want to understand how this process take place by measuring different scales and asking and trying to answer this question. So what is the contribution of VOC emission from soil, litter, and trees to the atmosphere? Are there predictable relationships among the scales, soil, plant, and atmosphere? And how do these processes mediated by VOCs change with forest degradation? So for the questions 1 and 2, we are doing our research in the ATTO site. So we do measurements at different scales. We have satellite retrievals, we have canopy fluxes of VOCs, we have leaf fluxes, and if you want to know more about that, please go to the poster of Michele, because this is her PhD project. We also have flux measurements of VOCs emitted by soil and litter flux, and this is the PhD of Deborah. She's not here today because she has something way more important in her life. She's about to become a mom. And Murieli is a former student from this group that investigated VOCs emitted during litter decomposition. And the idea is that once we have all the data from these different scales, we're gonna integrate and optimize the MEGAN, which is a VOC model. But for the question 3, that we want to understand how these processes take place in a degraded forest, we are going to do our measurements in another site, in the Tungurul Ranch, which is in the arc of deforestation in Mato Grosso State. And this is a very interesting project where they have permanent plots that, where they investigate forest recovery after a fire experiment. And these plots are also in border with an agricultural field. So the idea is that we are going to measure species in this gradient from the agricultural field board to the inner part of the forest and see how the emission of VOCs can change, because according to this conceptual model, we are expecting to have more species that emit VOCs that are related to temperature and drought stress, which is higher here. So in this figure, we can see the temperature, surface temperature gradient. So in the agricultural field, we have higher temperatures than in the forest. So we're gonna do these measurements first with drones. And if we have time, we're also gonna do some leaf level measurements with the dominant tree species that are in this gradient. And in the end, we will hope to integrate this data with the model that we have optimized ATTO. That's it.
Okay, thank you, Eliane. Okay, and now we have the next presentation for Michele.
So, hello, my name is Michelle. I'm a PhD student now for MPI for biogeochemistry, and I was a master's student at IMPA before. I'm also at the beginning of my PhD. I just finished the first year, so I still don't have any results to show. But my PhD is about plant traits and chemical diversity of tropical tree species in the central Amazon forest. And the focus is on plant VOCs, so biogenic volatile organic compounds. They were mentioned today before, so they are compounds involved in a wide range of ecophysiological processes, from cellular regulation up to biosphere, atmosphere, physical and chemical processes. And the Amazon forest can be considered the greatest and most diverse source of emissions because of its high plant biomass and high species diversity. Still, there is a lack of observational, particularly leaf-level studies that combined with the logistical challenges of measuring in these sites bring high uncertainty to the emission estimates for this forest. Meanwhile, foliar chemistry can be a possible driver of variation in both in chemical species and in magnitude of emission, but this relationship is not yet understood. And meanwhile, reflectance has been shown as a good predictor of leaf-stored BVOCs, phenolic compounds, and functional traits. And we know that leaf phenology and leaf flushing events are drivers of seasonal variation in BVOC emissions, and leaf chemistry changes with leaf age. So, in my PhD, we are asking 3 questions. The first being, how does BVOC emission capacity coordinate with phenological habits and leaf functional and chemical traits? The second, if there is coordination, is it possible to indirectly estimate the presence of emission capacity using NIR reflectance? And then finally, how are isoprene and monoterpene emissions driven by light and temperature in different phenological habits? So by understanding how emission capacity coordinates with leaf functional traits, chemical traits, and phenological habits, we expect to create a model that can indirectly predict emission capacity, the presence of emission capacity through NIR reflectance. And with this knowledge, combined with a deeper understanding of emission rates change with temperature and light in different phenological habits and leaf ages, we can advance BVOC remote sensing techniques and improve emission models. If you want to know more about this and the methods that I'm going to use, please come by my poster. Thank you very much.
Thank you, Michele.
So now we are moving on to the greenhouse gas, and we have the presentation from Helle.
Okay, hello everyone. So I'm presenting this as part of a team, like we are at the Max Planck Institute in Biogeochemistry, working with 4 postdocs on greenhouse gases. You see the names here, Santiago, Sam and me, and Komi is also, but he could not make it, but he is also watching online. So for any questions, feel free to write him or text him. So the first poster, today we have 3 posters. The first poster, I will just spend a few words on it, is just to show which measurements are going on at the moment in the A2 and the C2 container. As you all know, the greenhouse gas measurements have been paused for some time. There were some problems, but at the moment they are mostly all running again. So in the C2 container, at the instant tower, we have several measurements running. As you can see on the picture, we have picaros running. We have for water isotopes for different greenhouse gases. And in the A2 container at the TAL tower, we have an FDR newly installed. And also, more Carlos Scherer area, we have this beautiful ICOS sampler standing there. This poster is just to show an overview of which data we're currently sampling and hopefully also to keep on sampling in the future. And that's actually all about this poster. Please come to our poster for more information.
The next speaker is Santiago.
Thanks. Yeah, so as Hela said, in the group of Jena, in the group of Jena, we started doing some extra measurements around the flooded areas of ATTO. So the picture here is one of the plots of the MAWA group, where we did some tree stem measurements and very preliminary experiments in these places to get the isotopic signature, fluxes, and water methane concentrations to understand what I talked about in the morning, that we're trying to constrain this methane source and also to understand the variability of methane emissions around ATTO. So this is, this is a big effort. We found very interesting things, but it takes a lot of effort to get this type of measurement, so we are really wanting to share this with you, but we are also looking for people to help us, maybe some projects with the students. And so if you're interested, go to the poster, and what you see here in the plot is that there is a big variation of the stem fluxes of methane within a species, but also with height. So we measured at 2 heights of the stem. Generally, the higher measurement point has lower fluxes, and the lower measurement point has higher fluxes. But yeah, if you wanna check this more, so go to the poster. Thanks.
Our next speaker is Sam.
I think it's okay. So as Santiago was just saying, and as he explained this morning, we're interested in the sites that might be sources of methane to the atmosphere. So he's just shown you the data we took from the river. And in August, we also went to some of these valley sites in the plateau, these baixios, because they're wet, so they might act differently to the plateaus, right? You got the clicker? Just click. I didn't know that. That's all right. Neither do I. So we went, I can't really see this very well, but we went to a valley site at the end of Daniel Mara's Karabakh 3 plot, which is about a kilometer east of the ATTO camp. And similar to the flooded ecosystems, we looked at soil, stream, and stem methane fluxes. We found variation. Everything's about an order of magnitude smaller than the fluxes we saw in the flooded forests. But we also have some interesting variations with tree species and their role as sources of methane to the atmosphere. So the same thing again, if anyone wants to do a project on any greenhouse gases, we have too many instruments and too much work. So come find us.
So our next speaker is not here today, but she sent a video. Please, vocês podem ligar o vídeo, por favor? O vídeo.
Olá a todos, eu sou a Murielle e hoje irei apresentar um estudo que tem como título Compostos orgânicos voláteis biogênicos emitidos pela decomposição de folhas de serrapilheira na Amazônia Central, sítio ATTO, que foi desenvolvido durante o meu mestrado sob supervisão da doutora Eliane Gomes Alves. Bom, é conhecido que uma ampla diversidade química de compostos orgânicos voláteis biogênicos pode ser emitido pela serrapilheira em decomposição em diferentes regiões e que esses compostos possuem diversas funções dentro do ecossistema, mediando importantes interações entre solo, planta e atmosfera. No entanto, o que não sabemos é a contribuição desse compartimento em solos tropicais, especialmente dentro da Amazônia, floresta esta responsável pela maior emissão de BVUCs para a atmosfera. Bom, para buscar responder essa pergunta, instalamos um experimento de decomposição com litter bags próximo à torre instante, no sítio ATTO, área de platô. Foram realizadas coletas periódicas das folhas em decomposição durante 229 dias de experimento. E após as coletas, as litter bags eram encaminhadas ao laboratório e a partir de uma incubação eram feitas as medidas de VOCs, CO2, CH4 e análises físicas e químicas da folha. Como resultado, durante 229 dias de decomposição, houve uma perda de 31,4% da massa foliar, o equivalente a uma taxa de 0,72 gramas ao ano, ou 72% de massa perdida no ano. Identificamos 19 compostos orgânicos voláteis, classificados em dois grandes grupos, os compostos oxigenados e os isoprenóides. Também observamos que a emissão de BVUC diminuiu com o tempo e que a maior emissão foi no início da decomposição, em 20 e 38 dias. Além disso, encontramos valores negativos que indicaram o consumo de BVUCs, podendo sugerir que esses compostos serviram como fonte de energia para os micro-organismos, visto que nesse período também encontramos a maior taxa de biomassa microbiana. Não podemos deixar de mencionar que a sazonalidade é um fator importante nesses resultados, pois é conhecido que na Amazônia há uma grande deposição de folhas no chão da floresta no período seco, que é quando as árvores trocam de folhas, corroborando com os nossos resultados, pois já foi descrito que altas taxas de sesquiterpenos emitidos no período seco poderia ser advindo dos estoques de serrapilheira acumulado no chão da floresta nesse período. Encontramos um agrupamento das 3 primeiras campanhas em relação às 2 últimas. tanto para BioCis como para a maioria das características da folha, como nutrientes, conteúdo de água, foliar, lignina e celulose, sugerindo que os estádios iniciais tiveram maior contribuição na emissão. Então, esses são alguns dos resultados desse estudo. Quem se interessou mais pelo assunto, teve alguma curiosidade ou gostaria de entender mais os detalhes do trabalho, o pôster está disponível com a Eliane, é só procurar ela, e eu também estou disponível através do meu e-mail. Muito obrigada e um bom evento a todos.
Obrigada, Murielli. So we had contact with Ichiara and she sent us a video. So we have a last talk today of the missing speaker. So I'm happy that we have a last video. Pode começar.
I'm Mayra Albuquerque from University of São Paulo, and my work proposed to evaluate if carbon dioxide and methane cell products are adequate for Central Amazonia, compare them with 9 years of measurements took at the ATTO site. So I hope to find you at the post session Tuesday between 3 and 4 p.m. on our time to talk more about the select products adequacy for Amazonia and 9 years of carbon dioxide and methane ATTO time series.
Okay, so we've reached the end of our session. Maybe Viviana wants to take the word, or Sibeli?
No, I think now we just hang our posters and start the poster session. Outside. Yeah, the reason why it's outside is because we are not allowed to drink beer inside. So, and we know that we cannot avoid anyone of drinking beer, so we do it outside. It's either beer or warm environment, so.
Just a minute before we all leave. Najibi, Antonio, please. They were not here this morning. They were working to, to have our site working. So they missed, but now please, são para vocês as palmas. Vocês estavam aqui mais cedo. They wanna talk about something. They wanna talk about something. I'm just going to translate.
Boa tarde a todos. Eu me chamo Najib. Esse é meu amigo Antônio. Nós trabalhamos lá no projeto ATTO. E há uns 5 anos atrás começamos uma ideia também do nosso amigo Amauri e outros companheiros que estão lá também, fazer uma vaquinha entre nós e doar algumas coisas para a comunidade, para as crianças carentes. E assim começou o nosso Natal Solidário.
Yeah, so he just introduced, he's Najibi, Antonio, but like we all know, around 5 years ago, the idea started with Amauri as well, to collect some stuff to give to the inhabitants from the river during Christmas time.
Aí estamos pedindo a colaboração dos amigos e quem puder ajudar. Aqui o Pix, né? E esse também, para ajudar a gente a contribuir com a comunidade, que realmente os ribeirinhos merecem, que eles colaboraram muito com a gente no início do projeto e até hoje colaboram. São gente do nosso coração também.
So they are here asking for donations for this year Christmas baskets for the local community. They support the project as well, and even like them over there. So they like it a lot. They are all good people. So please donate. Those are the account informations over there in that banner. There is also there is also this box if you want to make some donations, like in cash. please feel free that they accept everything, like euro, dollar, like reais. There's
Obrigado a todos.
So there is a Pix account for the Brazilians. There is also a PayPal account for the foreign people, okay? So don't worry. If there is any question, you can talk to them in the field, or you can talk to any one of the coordinators, the administration here. So yeah. And now we are going to the poster session in person. And for the ones online, thanks for watching. See you tomorrow.